జాయిన్ అవ్వచ్చు ఆన్ చేశారు. చిలుకలు ఏం సౌండ్ చేస్తున్నాయి? చిలుకలు. దేనికి ఇద్దాం? ఎవరు చేస్తారు? సుమిత్ర మేడం లేరు. చెప్పండి.
CSIR Research Associate and Postdoctoral Researcher. And from 2018 to 19, Sir has worked as a postdoctoral researcher at University of Kansas, United States of America. From 2019, Sir has worked as an assistant professor, Department of Chemistry, IIT Delhi. Sir has received some awards and achievements like Travel Grant Award and Senior and Junior Research Fellow and MSc Entrance third rank from Kakati University. The research interest areas of Ramu Yadav sir are cooperative catalysis, carbon fluorine bond formation, and artificial metal enzyme catalysis. And sir has 15 publications in a reputed national and international journal. Sir has done 3 reviews and book chapters. Now, given many poster and oral presentations in national and international conferences. Now, I welcome Dr. Ramu Yadav sir to deliver a lecture, and over to Ramu Yadav sir.
Yeah, thank you, Dr. Gnaneshwar, for giving a brief introduction about me. And also, I would like to thank Professor Noorjahan for inviting me as a invited speaker in this symposium. And I think new normal nowadays through online. It's a great honor because I also studied my BSc degree in MBS Degree College, Mabnar. I know Mabnar very well. I roamed so much around that. So with further no ado, I can directly go to presentation. I would like to share my screen. Can you see my slides?
Yes, sir. Yes, sir.
Yes, sir.
Okay. Yeah, nice. Today's my talk will be mainly based on my postdoctoral work, where this has done at University of Kyoto University in Japan. So this mainly deals about transition metal catalyzed nitro cross-talking reactions. So for the benefit of master student, I have given a brief introduction of some their future opportunities. So let's start with something. So this can be considered both either any chemistry, either organic or physical. As I know organic more, that's what I prefer to show here. So once the student has did his masters in organic chemistry, if he has some fellowship like GATE or CSR, he will have two options. Okay, so he goes through MSc, like he can go to pharmaceutical industry, or he can go like DRDO or BARC, the public sector job, or like a teaching in private college or in coaching center. So what he realizes, if he go this way, he feel like life seems to be like monotonous, no something different. Uh, he will feel so bored after like 10 years or 15 years. Except few guys, there will be always exception, but most of them will be fed up. Like though there is no interesting. Let's see if somebody has interest on the PhD with this fellowship. He started doing PhD, then his life will be very excitement in terms of like happiness, and it also follows some kind of sadness. Means he will go definitely so much mental imbalances. Even he get success, he will be happy. If he get failure, he will go like disappointed. So life will not be like same. It will be a mixture of many things. So if some candidate student has done some PhD, let's say, he has two options. He can go to pharmaceutical industry. As you already, I think many of you already in good position and with a good growth and have some money you can earn in the industry. Let's say for some case, some students are very interested to pursue higher research again. He can go to a postdoctoral study with JSPS Humboldt Fellowship. He can earn very good amount of money. After postdoc, further he can go to the pharmaceutical industry or he can settle as a faculty. So both have their own advantages and disadvantages. If you want like money and also service, give employment to the society back or in terms of your own country or your own village, whatever it may be. So you can create job by going to pharmaceutical industry. If somebody, let's say, if somebody interested to go like a faculty, although he can earn some decent money, but here you will have a more flexibility what you want to do really if you want to do some innovation. So I think by this, whoever like master student, they should decide their life at the time of when you are finishing your MSc. So whether you want to take this path or this path. Definitely every path has a different kind of success and failure. You have to overcome all those things. Then your life will be much more meaningful. So I will give special examples how somebody, if we did PhD and enter in a pharmaceutical industry, let's consider like Dr. Anji Reddy. I think it's very well known. He's one of the pioneers to establish pharmaceutical industry in India. As all you know, Dr. Reddy's pharmaceutical company. It is one now worth of like $3.2 billion company. So he did his PhD in organic chemistry from NCL Pune. So it's a one example. Let's take our own, who can proud of like Dr. MSN Reddy is from Palamuru, right? Everyone knows. Now he's now like MSN founder of MSN Laboratory. He's, I think, it's one of the fastest growing pharmaceutical industry, I think, in India. Even you can consider after a few years in old also. So you should get motivation like this. Even you can take Lawrence Labs. He's Satyanarayana Chava is from Andhra University, PhD. So what I mean to say, think in a big, in terms of big, because you will have a lot of plenty of opportunity. So I hope maybe from the down the time, like 10 years from your university, I should expect by motivating some good position. So this for motivation. Let's come to our main topic today's lecture. So as here I shown, you can see most of the molecules contains biaryl ring. So you see this biphenomycin B, AMG 837, other boscalide and CBP. These all contains are in moiety as a core. And these molecules have a wide importance in like pharmaceutical, organic molecules, and even agrochemical. So this is a very important unit for as a active moiety in many fields. So that's why different scientists has focused to develop very good methods to construct such biphenyl rings. So you all, let me, you all know that how this can be synthesized basically from cross-coupling reaction. It is one of the best innovation in catalysis happen, like coupling of aryl halides with aryl boronic acid in presence of metal catalyst can lead to the biaryl synthesis in very structural diversity and also with high efficiency. Here the aryl halides acts as a electrophile and the aryl boronic acid acts as a nucleophile. So you are coupling both electrophile and nucleophile with the help of metal catalysis. This name cross coupling comes because you are once this aryl electrophile undergo oxidative addition with palladium complex, another organometallic is cross coupling from boron to metal center. That's why it's named as a cross coupling reactions. This part is fine, very good. So if you want to access the aryl halides, generally it can be accessed from the diazonium salt via Sandemeyer reaction by treating with copper chloride. Okay, so you need one more synthetic step from diazonium salt to prepare aryl halides. That's why different groups have tried to develop methodology from directly coupling with the diazonium salt with the organometallic reagent of like boronic acid or saline reagent to make biaryl ring. But this was not gain much popularity because of different shortcomings like not good substrate scope or not tolerable in total synthesis. So if you in turn look to prepare diazonium salt, you have to come from nitrobenzene via reduction to amine, then diazotization. So it means from nitrobenzene, you need 2 additional synthetic steps to prepare aryl halide electrophile. So this, if you consider the industrial point of view, it's a huge cost and environmental, it's a huge, you are generating a very hazardous chemical in a large quantity. So then we wonder, because cross coupling has developed like 6 decades ago, but no one thought of this such transformation. So we thought, why don't we can develop directly by using nitroarenes as the electrophiles in coupling reaction. So this was our idea to develop a new route for biaryl synthesis. So in 60 years, nobody developed, means there should be a lot of challenges in this transformation. Let's see, like there is no catalytic system properly to couple nitroarene to the aryl halides. So we initially have to come up with a new catalytic system. The another big challenge is to cleave the carbon nitro bond, which is very strong. So you need a lot of electron rich metal complex to undergo oxidative addition. The other problem with nitrobenzene is nitro group is a oxidant. It can poison the catalyst or it can easily undergo reduction process to convert to the aniline derivative. So with these very challenges, I think people have not focused on this transformation. So we have taken up this challenge. So ultimately what we are going to achieve by developing this transformation is we are offering a new electrophile to the synthetic community or to industrially. So these aryl nitro compounds are commercially very cheap than the aryl halide. So we are also providing a cost effective method to synthesize biaryl moiety. And also good thing is that you can modify the nitroarene very easily compared to the aryl halides. I will show in the next slide. So as this is our desired reaction, you can easily understood, right? If any halo group on the electron withdrawing nitroarene, you can easily displace that halo group with different nucleophiles via aromatic nucleophilic substitution reaction. Or even you can also do like ortho or para CH functionalization with simple base or metal catalysis to decorate this nitroarene very easily. So this is not possible with aryl chloro compound. So these are one of the biggest advantages in aryl nitro compounds. So once if you can decorate these nitro arenes easily, if you're able to develop new coupling reaction, then you can introduce more complexity on this aryl moiety, so which can eventually find big application for pharmaceutical or agrochemical. Even you can also incorporate the material chemistry also. So with these advantages, we check the literature, what are the possibilities are available. So for good news is that when we check the literature, we found some substrate biased nitro group living. As you can see, a Sujitrose type of reaction where allyl nitro compound, when treated with palladium zero complex, it happened that nitro group has left as acting as a living group to form pi allyl palladium complex. So once this pi allyl palladium complex is generated, that you can easily attack the nucleophile. So this is one example has been reported. In other case, where they have taken like highly electron deficient 1,4-dinitro compound, which treated with trialkyl boron in presence of p-butoxide. In this case also, it gave replacement of nitro group as a living group to replace with alkyl, but at the cost of many side products. So this is synthetically not a very good method to replace nitro with alkyl group. So this type of reaction has proceeding via radical mechanism, because this is highly electron deficient, right? So it can take proton from the p-butoxide anion to make a radical anion type intermediate, followed by the p-butoxide radical can attack and leave the R radicals, followed by the reaction with the anion radical type of 1,4-dinitro benzene can result this alkylated nitrobenzene derivative. And later there are few reports with exclusively with electron deficient nitroarene as employed for the CO and CC bond formation, CO and CS bond formation in presence of rhodium complex and copper complexes. Here, this cleavage is much easy because you are polarizing the carbon nitro bond by using the electron withdrawing group on the arene moiety. So such substrate is very easy. If you put methoxy group here, the reaction will not proceed. So this is a very substrate biased C nitro coupling reaction. As you can see here, both all are CS, CO, and also such alkyl is reported, but they lack substrate scope and there no reports for biphenyl formation. With this background, we have screened several catalytic combinations. Here I have briefly summarized our whole result. We have screened almost like 500 to 600 reactions to get this reaction condition. Okay, so as I mentioned, we have took very strong, challenging substrate, that is methoxy nitro, because as you can see, this is withdrawing group, right? Nitro. If it is electron donate, it becomes much more strong to cleave the nitro bond. So the idea is, if we can able to develop this very challenging substrate, this can easily applicable for electron withdrawing substitute nitrobenzene. That's the idea. With that idea, we have took this substrate and screened several catalysts and found with palladium ECOC in 5 mole%, and I will show in the coming ligands in 20 mole%, and we also employed this 18 crown 6 in 10 mole%. This acts as a phase transfer catalysis. Okay. K3PO4 as a base in diaction solvent at 130 degrees centigrade for 24 hours. So these are the summary of best catalytic best ligand system. As like S4, you can see here, this is a bi-aryl monophosphine ligand. Such type of ligand has shown very good reactivity. This is, you can attribute that when the palladium coordinate with the phosphine ligand. So when it reduced to palladium zero, this is there, right? This can coordinate, easily trap the palladium zero to become dead of palladium complex. So this is making more active palladium complex rather than monophosphine simple aryl phosphine ligand. So when, as you can see, we have more screened several such similar type of ligand. With Rufose, we got 15%, and when we use X4 around 56%. When we introduce and expose some electron donating group like methoxy, the yield was increased up to 86%. Here, a couple of points to highlight that. So you can wonder why this base K3PO4 NH2O is very important. Why not just dry K3PO4? So the idea is water acts as a base to make this boronic acid more nucleophilic by coordinating here OH. So that you call it a borate species, means this becomes a more nucleophilic in nature. Okay. Next case, why this 18 count 6 is important means this is inorganic base, that is K3O4 is inorganic base. So to bring it to the organic solvent, we need a proper, so 18 count 6 is very good for trapping of potassium cation so that this can come into the solvent to accelerate the reaction. So I hope you clear with this why we used. So with this Bedford's ligand system, and these are the combination, we got 86% yield, and we any reaction to validate the methodology, we have to show very good substrate scope. That's why we what we next screen, as you can see here, initially electron donating like 4-methoxy or 4 toluene or phenyl morpholine. You can see gave a very not very good yield, but moderate to good yield, like 63% to 76% yield. And even phenyl also is okay. It's a pi extended tether nitrobenzene. And you can see even electron withdrawing like aldehyde or keto has shown also very good reactivity for in this coupling reaction. Only problem was like fluoro compound has not tolerated very well. Only we got 46% yield. You can further some substrate, like I would like to emphasize on this ester or sulfonyl. These also gave better yield. But the highlight point, key point is that when the steric hindrance incorporate near to the nitro group, the yield has not decreased. The yield was maintained. This is one of the highlight of this catalytic condition, because in general coupling reaction, when you introduce steric bulk near to the any coupling center, the yield generally used to drop because of the steric hindrance. But in our case, we did not observe. Even pi electron rich compound also worked well. Even heteroaromatic nitroarene also, as you can see, pyridine and quinoline, isoquinoline has very well. So overall, the scope is very good. I cannot say very broad because there are many limitations with this scope, but that is fundamental, fundamentally to show the nitrocoupling reaction, we have shown that. Even with respect to the boronic acid scope, like nitro with electron donating N and dimethyl aniline derivative, or even toluene 4-tertiary butyl also okay with this condition. Even electron deficient like acetyl or ester based, you can yield is moderate, not very good yield. The problem with the boronic acid, we could not achieve best substrate scope with heteroaromatic boronic acid. Apart from that, all worked very well with this catalytic condition. So once established the catalytic condition, then we have shown the broad utility of different like nitroarene and also boronic acid. Then we look to how can we enhance this methodology by finding some different application. That's what we did. So it's immediately in our mind, it appeared that what happens if 2 nitro group as a part of molecule. Okay. So whether it 2 nitro immediately participate in reaction, or we can do the sequentially. So to our surprise, and also to happy that it we stop reaction after short time, like 3 hours, we are able to selectively annihilate only 1 nitro in 61% yield. Then the next nitro has been subjected for another boronic acid to introduce 2 different aryl moiety on the same molecule. So it's a very, I think, nice demonstration to show sequential coupling reaction. As you can easily imagine that it's a very high electron pi rich system. Such molecules will, hello? Is it okay?
Okay, sir.
Somebody didn't know disturb it. Okay, so you can easily imagine these kind of molecules have wide importance in material chemistry, like OLED type of systems. So later, we have also examined, as I initially indicated, if any halo substitution on electron nitroarene, you can easily displace this fluoro with base in presence of base and nucleophile. Like that, we introduce carbazole and phenol in very good yield, around 82% and more than 90% yield. It's a very simple reaction. Then we subjected to our catalytic condition. As you can see, we achieved the nitro coupling in 59% and 67% yield. So this such decoration on the arene moiety not possible with chloro. or hydro or bromo. So this shows the complementary approach of nitroarenes in the cross-coupling reactions. Even you can selectively functionalize if simple ortho and para CH bond by using, in case of ortho CH bond, we can do it palladium catalysis, or if you want to do functionation of para, you may know, right? This is VNS, vicarious nucleophilic substitution in 80%. So such complexity introduced nitroarene also underwent very successful reaction with our catalytic condition 52% and 71%. So this shows how broad utility of nitroarene electrophiles over haloarenes. So this is very good actually, but there are, as I said, there's many limitations are associated with this methodology. As if it is any chloro or halo is there, like instead of fluoro, if you subject this condition, it first basically because of more polarity of this halo bond, initially the coupling reaction will undergo on chloro or bromo or iodo. Like that, many substrate has not compatible with this present catalytic condition, or even like haloboronic acid, or as I indicated that this heteroaromatic boronic acid also not compatible with our catalytic condition. So these are the one of the disadvantage with this method. After we have seen we have developed a methodology, substrate scope, and limitation. Then we want to, as a fundamental science, we want to study the reaction mechanism, because people have so far no one showed this oxidative addition of nitro RL nitro bond. That's why we undertook such a task to unravel the mechanistic details. So as you can see here, this has been done under NMR experiment. So what we did was we have mixed all this catalytic condition in the NMR tube. This is especially Jeong NMR tube for the benefit of MSc or PhD students. So NMR experiment will be done. So this Jeong NMR tube will be introduced. This is called as a glow box. So we will mix all the substrates in this NMR tube in glow box. Then heat it outside around 60 degrees for 5 hours. Then subject it to NMR in the P31 NMR, because we are monitoring P31, which is easy compared to the monitoring proton or carbon NMR. Okay, so we distinctly observed two peaks, like 37.8 ppm and 48.2 ppm. So this actually, then we have become very curious, what could be these things. So here I've directly shown you what is the intermediate of this. In the next slides, you can see we have done also stoichiometric experiment. So the peak corresponds to the 48.2 belongs to the nitroso compound. That means one oxygen lost from the nitro group. That resulted the pi code by this NO double bond coordination of the metal complex resulted at 48.2. The peak result correspond to the 37.8 is correspond to the oxidative addition of nitrobenzene. So these are very, very, I think, one of the interesting complex in the organometallic chemistry, because in generally people thought this nitro group as an oxidizing agent, so it mostly kills the palladium catalyst. So this was also, we have done some mass analysis also, that there also we can able to identify this corresponding peaks, like oxidative addition of, because this in the mass that NO2 has lived, that's why we got the cation complex. And as you can see, nitroso compound has been shown in this. It is mass also confirmed. So later we want to check exactly, we subjected this, see, especially the palladium easily, you can generate palladium zero complex from this easily. So when we treated this palladium complex with nitrosobenzene with the Bedford's ligand in toluene, we able to get this nitroso compound coordination to the palladium complex. You can see in the crystal structure. So this was when we recorded P31, it corresponds to the 48.2. So as I told you, right, this 48. So this we have confirmed this P corresponds to the nitroso compound. And to our delight, when we the similar complex treated with nitrobenzene, one equivalent, we are able to get around 28% of oxidatively added nitrobenzene and this was further confirmed by the crystal structure. This is the first oxidative addition complex of nitrobenzene and palladium complex reported in the literature. This is corresponds to this 37.8 ppm peak. So after establishing the oxidative addition complex, so we have to still study many aspects of this transformation. So what we did was, we did some stoichiometry experiment. Is it really true intermediate in our reaction? To confirm that, we have subjected this oxidatively added complex with boronic acid in presence of K3O4 dioxane, which resulted 92% of biphenyl formation. So this indicates that this is actually very crucial intermediate in our transformation. Same thing was further confirmed by this catalytic conditioner, so where we use this as a catalyst. So even you can see here around 72% yield. So that clearly establishes this oxidative addition is happening in our reaction. So later, to rule out some, is there any possibility of in situ generated nitro or nitrous as an intermediate? When we subjected this substrate, we could not see any product formation. This ruled out these are not intermediate in our transformation. Even we have ruled out this radical type of intermediate by introducing like tempo or galvanoxyl reagents. We could not see any, the yield is down, but product formation is happening. So this also ruled out the possibility of radical intermediate. So maybe the low yield, because these are the oxidizing agent, it can oxidize this bread force path-spin ligand. So that's why reaction has decreased. This has given some clue, but further we don't know what is the rate determining step, right? So to that, we have carried out some kinetic experiments. So in the kinetic experiment, we will vary the concentration of nitrobenzene, boronic acid, or palladium complex, and also ligand. So by changing such concentration, we can understand who is controlling the reaction. Okay. Further, initially we conducted the kinetics of nitrobenzene. So as you can see, we found this non-integer like 0.3 or 0.5 order. So which is little confusing. It's either 0 order or first order. So this I will explain later. If we at this stage we have considered a 0 order, that means it does not depend on the concentration of completely on the nitrobenzene. It's a completely 0th order, and boronic acid, as you can see, we identify the slope is 0. So that means the rate of the reaction does not depend on the concentration of boronic acid. Okay. So later the same thing has conducted with the palladium complex, that is bread force palladium G3. You can see easily first order reaction. It should be, because without catalyst the reaction will not proceed. So first order reaction with palladium complex. With the bread force, it observed 0th order. So the concentration of if you even increase this 35 mole % to even 40, reaction does not accelerated. So to account why such integer of value we got with nitrobenzene, we assumed that so there is some kind of resting state in the reaction medium. So like it is known in the literature that if any like such substrates are there, it can easily bind to the palladium complex through the pi coordination. So when this happens, this has to undergo oxidative addition, right? That is, I think, rate determining step, because this needs more energy in the reaction medium, whatever we are heating 130 degrees centigrade. So, and most of the ones, this complexes also very easy. When we mix palladium complex with nitrobenzene, we can get this resting state palladium complex nitroarene substrate. So to further assess this intermediate, we have we are happy to isolate with pi rich this 1-nitronaphthalene, where you can see the pi complex, the nitroarene with palladium complex. This has been further confirmed by the X-ray analysis. So this gives a very complete picture. What it tells us that before undergoing oxidative addition, there is some resting state with pi complex of nitroarene with palladium complex. That's why we are getting a non-integer value for concentration of nitroarene. So by combining all this data, we now concluded what is the actual mechanism in this transformation. As you can see, once palladium 0 can easily generate, so which initially forms the pi complexes with nitroarene. So from pi nitroarene to the oxidative addition is rate determining step. Then followed by the transmetallation, which is very easy step. So it does not need any much energy. So once nitro, after transmetallation, it can easily undergo reductive elimination to produce this biphenyl formation. Again, you get back your palladium active system for next cycle. So this, whatever we found this rate determining step on the oxidative addition is very important in this reaction. So once after establishing this Suzuki-Miura coupling, we next also easily applied this conditions for Buchwald-Hartwig coupling, and we can also successfully achieve the CN bond formation in 83%. We have shown in 20 example in this paper. So you can see most of the catalytic system is similar like Suzuki coupling reaction. Later, this was extended for reductive denitration. Generally, you people know, right? If when you put any reductant, nitro group undergo to aniline formation. But with this catalytic condition, we are able to cleave selectively the carbon-nitro bond by using simple isopropanol as a hydrogen source. This was further confirmed by this deuterium labeling experiment. As you can see, that this one will be incorporated here in greater than 97% yield. So overall, this I think one of the challenging transformation we have succeeded. We feel that this is not the best condition. This actually gives a very fundamental insight of this transformation. I think better catalytic condition needs to be evolved later stage to incorporate this methodology towards the industrial application. But we believe one day this will become a textbook reaction. So this is my, so far I explained my previous where I was like a co-worker with some supervisor. So this I want to do at IIT Delhi, and also you can think like a future plan. Our major focus is like development of new concepts in the catalysis, like in terms of development of new reaction, and also to understand what is the inside flask is going in the solution. Actually, that is very important. By knowing the mechanics studies, you can develop very, very applicable, like industrial applicable catalytic system. And later we want to extend this methodology towards the biomedical or natural product synthesis based on our own methodology. We don't touch not like a regular total synthesis guy. So I can give you a brief overview what we are looking here. I don't know why. So in the catalysis, like homogeneous catalysis, our major focus is on to develop new carbon-carbon bond formation by using readily available carboxylic acid, either aromatic carboxylic acid or aliphatic carboxylic acid. And also we are particularly interested to develop new methodology for carbon fluorine or carbon trifluoromethyl bond formation. So because this carbon fluorine incorporation in the drug candidates can improve the lead lipophilicity, metabolic stability. So it's a very, I think, need very good methods for industrial application. And later we also, another area on asymmetric catalysis, we want to develop new asymmetric catalysis based on the two approaches, like artificial metalloenzymes. That means we mimic the nature metalloenzymes like cytochrome 450. Like this, we want to replace iron with palladium or nickel and develop new catalysis. And also we will focus on new ligand design for asymmetric synthesis. So later we want to employ our methodology towards the new bioconjugation method, or we would like to incorporate this fluorine bond in the existing drug or new drug to enhance their activity or completely find a new reactivity. So these are our overbroad view of research at IIT Delhi. So overall, we want to develop like new innovative and also cost-effective commercially viable synthetic methodology for the synthesis of pharmaceutically relevant molecules, and also like it's a majorly for functional molecules to synthesize, develop new methods. So to pursue these goals, currently we have two PhD students like Ruchi Sharma, she's from Kurukshetra University, and one other guy, Suresh Chaudhary, is from the University of Rajasthan. And also currently we have two master project students like Surendra Ram, Amar Prasad Majid, both are from IIT Delhi. And we got recently funding from as a startup fund from IIT Delhi around 20 lakhs and DST India from around 30 lakhs. So we are very grateful to them to support our research. And I would be very grateful to my supervisor, like PhD mentor. I think he's one of the inspiration. He always motivated me whenever there are some difficult situation. Like I have a very good interaction with Professor Akios Kumar Saho, and I learned many things from Professor Yoshia Kinako from Kyoto, because how to analyze each. Even if you don't get any product, if you get side product, you have to give care to the side product. From there you can learn many things. So I learned from this Professor Asia Kinako. I'm very thankful to them. And the fluorine, some basic chemistry I got from Ryan Altman. He was at that time Kansas University. Now he's at Purdue University. And also my PhD fellowship was supported by CSR JRF fellowship. So I'm very thankful to CSR JRF, CSR India, and also DST for my PhD project funded, and CREST for my postdoctoral funded. So I would thank for every one of you for giving your time and attention for today's lecture. Thank you. I'm happy to answer any questions.
Thank you, sir. And thank you for your scintillating lecture on cross-coupling reactions, palladium-mediated cross-coupling reactions. Now I request participants, if you have any doubts, you can unmute and you can ask questions.
Dr. Ramu Yadav.
Yes, sir.
It was very nice presentation on your catalysis, and it is really impressive. And this is just comment. And we have two common features between us. You know, you are from University of Hyderabad PhD, and I am also, but at different times, 1994. But you did postdoc at Kansas, University of Kansas.
Yes.
I did postdoc as well, but we were different times with Professor Gunda Garg.
Oh,
So I don't know Gunda Garg. So she must have left by the time you are there, I guess.
Yeah, I think, yeah. She's in medicinal chemistry. I was in medicinal chemistry department.
Yeah, I'm in the medicinal chemistry Lawrence campus.
Okay.
So, and she left to the Minnesota for a medicinal chemistry chair.
Yeah. Now I can remember. I think she's one of the top professor in the department. People used to tell us.
Yes. That is nice. Interestingly, you are from Palamuru as well, and then two PhD and postdoc commonalities between us.
Yeah. Yes. Any comments is good to improve.
Yeah, it means it's a very nice presentation, and of course, very interesting is I will kind of tell you presented first two slides, Dr. MSN Reddy, MSN Labs from Palamuru. Incidentally, MSN is myself, is in the from same village. We are neighbors and we are cousins.
Very nice. Very good.
MSN is your cousin, huh? Okay, nice.
Yes.
Yeah, students can ask something.
Yeah, the session is open for discussion. Anybody can ask the questions. So if there is no question, then we will move on to Dr. Chandrakiran to have a concluding remarks on presentation, today's presentation.
Good evening to one and all. It's great pleasure for me to give concluding remarks regarding the talk given by Dr. Ramu Yadav, sir. It's a wonderful lecture, sir, very informative to our students. He clearly explained how simple substrates like nitroarenes or nitro compounds, how they are transformed into new carbon-carbon bond forming reactions, and also new CN bond forming reactions, and also reductive reactions to convert into simple arenes, reducted arenes. So these are very simple, looks like very simple reaction by applying various palladium metal catalyst. So all these reactions are there in our syllabus also. Such reactions are easily done at the laboratory level and published in reputed journals right? So it's really, it's a great sir, and great demo for students also, because it's very useful, and they will inspiration from this, because these compounds are easily available on the bench. So this can be transformed into very useful compounds. So therefore, this lecture would be very useful for our students. Thank you, sir. It's a wonderful presentation. We look forward for your.
Yeah. So actually, I forgot to say one thing. If you are some of students interested to pursue PhD, any of our department, they are welcome to contact us.
Yeah, definitely, sir, because we are having every day, every year, we have fellowship students like Inspire Scholars are there, and CSR fellows are there. So we'll definitely forward our students at your reputed institute. Like in the morning session also, one of our professor has told the same thing. So please send your students to the IITs for the further doctoral studies, so that they can have a better exposure and research work and have a great career in their life. So thank you, Ramu Yadav Garu, for joining us, and within a short notice you have joined me and joined us, and you have extended your cooperation readily. And one more thing I just want to convey on this platform. Actually, I scheduled this for 2 days actually, but because our PG examinations have been started today, so I just concluded to 1 day. So I just requested Ramu Yadav Garu to shift to today's session. So he readily accepted. So sorry, sir, it's very uncomfortable to you, but you have extended your cooperation. Thank you very much.
That's it. No problem. Thank you for giving me opportunity
Yeah,
for this.
one more,
Yeah,
sir.
on my behalf, as a senior professor in the department, thank you once again, Ramu Yadav Garu, for your excellent, wonderful lecture on cross-coupling reactions, and you have replaced the traditional conventional reactions where diazonium salts are used in the synthesis of biaryl compounds. And in fact, these reactions, they're very much useful to the MSc students. So on this platform, I'm requesting you one thing. So because as you said, you are also from Mehboob Nagar, you have done your, I think, degree at MVS College. So
Yeah.
you sometime whenever you try to visit your place, we invite you. Please let us know so that we invite you for an extension lecture, so that the students more they get inspired by people like you, because the students definitely our students especially, they think all the time that we are very less to take up higher studies or higher choose higher careers. So definitely, when our students see the people like you, they definitely get inspired and motivated. So the chemistry should not end at graduate or postgraduate level, isn't it? At a
Yeah.
graduate or graduate level, it has to extend, and that too, it is a university. So if people like you contribute, and we have also invited the morning speaker, Dr. Janakiram. So please do make it convenient, sir, whenever possible, whenever you are coming. So you please, like, we'll also be in touch with you, and we make use of your services as and when. And as you said, yes, definitely, we do motivate or forward our students to pursue PhD in IITs. And not only that, whenever we require any help with regards to any assays or any analysis with regards to, because ours is a budding university, we may not have all the facilities. So definitely we look forward, sir. Thank you.
Thank you. Thank you. Definitely, I will make a visit if I will coming to the hometown.
Thank
Yeah.
you.
One more thing, I requested Dr. Sudarshan Reddy to present a certificate of appreciation to Dr. Ramu Yadav. Edit.
Thank you.
Thank you, sir. We'll send this certificate to through mail, the signed copy. So thank you. It's a very less words for whatever you have done today. So I look forward many more interactions with you in near
But
future, and
you,
definitely,
you
as
should
Adam said,
motivate the students to participate in the interaction, because otherwise there is no point of presenting white
yeah, yeah.
chemistry, right?
Yeah, yeah. Actually, our students, you know that they are very shy and are afraid of such
No,
platforms to ask.
yeah, we are also
So,
like them,
yeah,
right? When we are doing MSc,
yeah, yeah,
you know,
yeah.
here in
They have,
IIT student, they will grill the professor like anything.
I know.
yeah, that's the difference between the state university and the
That's
Because see,
IIT.
what
what,
actually we have arranged. The reason why, the main reason why we have arranged this webinar is today's speakers, all the speakers, they have shared their research work. So even though it is not just not little, it is beyond their scope of understanding, but still we made it a point so that at least they think the chemistry what they are learning at MSc is not simple, and in fact that can be extended to any level, and so that they can reach to any heights. So that's the main reason we have invited all eminent speakers, and that's how the students get motivated. But at PG level, definitely the students who are really very shy also, they can really prosper well, and they can really do well after completing their PGs.
yeah,
And the only thing is they should constantly get motivated by people like you.
definitely, definitely, will help sometimes. Come and discuss with them.
Yeah. Thank you.
Yeah.
Thank you, sir. Now we will move on to the next lecture by Dr. P. Ravi Kumar Garu, Associate Director, CMC, Vanda Pharmaceuticals, Washington, DC, USA. So,
సుదర్శన్
welcome,
Yes, ma'am.
అందరినీ మిగతా వాళ్ళని మ్యూట్ చేయి, డిస్టర్బెన్స్ వస్తుంది మధ్యలో.
welcome Dr. Ravi Kumar Garu.
Madam, thank you for inviting me to this event, and it's always a pleasure and a more happy moment for me to come forward for this type of occasions, especially for the university from our own Palamuru district, and it's a honor and privilege to be part of this event. And I know, Rija Madam, she is our professor in AB College when we did our MSc, and most recently when we got connected back, she has asked me to and present a lecture for the university students. And so
Sir, before you start, excuse me, sir. Let us introduce you to the audience.
Sure.
I request Sudarshan Reddy to read out the profile and introduce the speaker to the audience.
Thank you, Noorjahan Madam, for giving me this opportunity to introduce Mr. Ravi Kumar P. On this afternoon session. Mr. Ravi Kumar P. born in a small town of Bhakti. Now presently it's a district now. He studied in a government school in a remote village, Marchal, until his 7th standard. He developed passion for chemistry during his intermediate years. He then went on to complete his MSc from AV College, I think under the headship of Girija Madam, affiliated to Osmania University. He worked for Dr. Reddy's laboratories for 3 years before going to US for his doctoral program at University of Missouri, Columbia in radiopharmaceutical chemistry. His research area was synthesis of phosphorus-based ligand systems for complexation with technetium and rhenium metals, and synthesis of radioactive gold nanoparticles for medical diagnostic applications. He has published several publications in reputed journals, including Journal of American Chemical Society, and presented his work in national conferences, including Society of Nuclear Medicine. After graduating, he worked as a radiopharmaceutical scientist in Department of Radiology at Washington University in St. Louis Medical School, a world-renowned institution which has produced more than 20 Nobel laureates. He then moved from academic research to industry and worked on the development of several new drugs in the CNS and oncology area. He is currently an associate director, CMC, R&D at Wanda Pharmaceuticals, located in Washington, D.C. Some of the drugs he worked include Hetlioz. Uh, Hetlioz is first and only medication that has been proven to treat people with the non-24 disorder. It has also completed phase 3 clinical trials for treating sleep problems in children with Smith-Magenis syndrome. Next, tradipitant is an NK1 receptor antagonist, which is in phase 3 clinical trials for 3 different indications. Those are atopic dermatitis, gastroparesis, and acute respiratory distress syndrome. In severely ill COVID-19 patients, which we already, I think Sir already told during the felicitation program of Girija Madam. And now, and the third one is trichostatin A. Trichostatin A, a potent HDAC inhibitor, is currently being developed for treating hematological cancer. Ravi sir has extensively worked on development of synthesis, procedure, preclinical evaluation, and the submission of an investigation new drug applications IND. The FDA has approved this IND and it is in phase 1 clinical trials. Apart from this professional life, he lives in Washington, D.C., sub-area. He enjoys traveling, cycling, swimming in his free times. Now I request Ravi sir, please, you can start your lecture, sir.
Thank you, sir, for your nice introduction. Is it audible now?
Yes, sir.
Yes, it is audible, Ravi. You can proceed.
Okay, yeah, sure, madam. Yeah, I'm just starting my slides. So as Dr. Ramu Yadav was presenting his presentation, he initially gave some options for the organic chemistry students, especially for chemistry students overall. Several options like academic research and pharmaceutical industry. So being in a pharmaceutical industry, I thought for students to, most of the students, the learning process is all it involves the chemistry and research areas, but a systemic approach to drug development is not, it doesn't give a comprehensive understanding of systemic drug development process during the master studies. So I thought should present a brief overview of the drug development process so that people who choose that career after their PhDs or masters, so they have an understanding of the process, what it takes for a drug from taking from the discovery stage to commercialization of the drugs. So what is drug development process? The how it started, and so the first systemic and rational synthetic drug development process actually started in early 1900s, and it was mainly by Paul Ehrlich and John Langley and others, elite scientists in the medicinal chemistry field, who carried out a systemic approach, by introducing the theory of receptive substances. And Paul Ehrlich, he was awarded Nobel Prize in 1908, and he was also considered as father of modern chemotherapy. And after that, in 1950s, the quantitative structure activity relationships was introduced, and then introduction of molecular modeling and combinatorial chemistry, the drug development process advanced much faster coming into the 20th century and 21st century. So usually for a new drug to come from the laboratory discovery stage to a commercial market, so it takes about 10 to 15 years of research, and it involves several $1 million, several $100 million to $1 billion to bring one new drug to the market, and also 1 in every 5,000 to 10,000 compounds that are evaluated actually reach to that stage. So the enormity of the research and work that goes on to this process is actually what I just want to give a brief introduction so that students can understand what involves in this process and what is the ultimate goal that research of these researchers who are working on the new drugs. So the main goal of any drug research actually starts with the disease. So you identify a disease which has unmet medical needs, or the disease could be already an approved medication is available, but you bring alternative options and alternative medicines to those diseases, which may have less side effects or more potent or more, which may show a more efficacy towards the disease. That's one goal, and then understand the disease and then identify right targets inside the body, these targets that could be a biological molecules or receptors or peptides, anything, and then discover the right molecule and that has some affinity towards that target that is chosen for a particular disease. So once it is chosen, and test and optimize that lead molecule to achieve a desired safety levels through animal studies and toxicology studies, and then conduct clinical studies in humans to evaluate the safety and efficacy of those drugs, and then get approval from the regulatory agencies. So usually the drug development has two arms. One is the discovery arm, and the second is the development arm. So feel proud, all chemists. So you are involved in both arms of the drug development process: the chemistry, manufacturing, and controls. This is whether you are in discovery or development, throughout the life cycle, the chemistry is always involved. So coming to the first arm of the drug development process, which is discovery. So there are two types of you choose a molecule in the discovery stage. One is structure based, so where the structure of the compound is already known, and it is already known to have certain pharmacological activities, and then knowing that it is further refined and optimized through toxicology studies and then it can take into preclinical activities for safety evaluations. So from that stage onwards, the structure based and the next is target based, and structure based and target based are common pathway from preclinical to post market. The second is the target based. In the target based, first in this strategy, you select a drug target. You identify a drug target inside the body. So these potential targets could be a receptor and a critical enzyme or another biologically important molecule So in the disease pathway, and once you identify those targets, you validate those targets, and then you take, once the target is validated, it is the next step is to identify a chemical entity which can interact with the target. So understand the chemistry of the target and then develop a set of lead compounds that can actually interact with the target. And then that lead can be further optimized to achieve desired safety levels. What is safety level? Should not be toxic to humans. So, and then it should have a good affinity towards the target. So once you achieve and optimize that lead, and then you move to the preclinical phase, which is the development phase, which I will talk later in the talk. So coming to the target-based approach, so these are the main steps involved in a target-based drug development process. So first you identify the disease, and which I already went through this entire cycle in the previous slide. And then, I don't want to repeat it again. So you identify the target and validate the target and choose some lead compounds and optimize the lead compound and and identify the right final lead candidate, and then take it to preclinical and then clinical to the market.
Dr. Ravi,
Yes.
you did not share the slides.
Oh.
You can share your slides, sir.
Yeah, actually, I do, but for some reason, okay.
Yeah.
Actually, Ravi, we forgot that you didn't share the slides because it was like a continuous story. We are listening to it. It's so interesting. So we never realized. In fact, thank you, Hari, for reminding for the benefit of the students.
No problem, because
Sorry, is everyone can see the slides now?
Yes, sir.
yes.
We can just have a slide. Yeah.
Yes, yes.
Fine, sir.
Just you just go through all the slides once. Just
Okay.
move quickly so that students will be benefited.
Okay. So in the first slide, actually, I showed the how the drug development actually started, and in early 90s and 1900s, and uh, the introduction of the theory of receptive substances, and then the development of QSARs and molecular modeling and docking and combinatorial chemistry, how that process has actually advanced the drug development process much faster in this century. And there are many unmet medical needs or have now treatment options available after these advances in the combinatorial chemistry and computational chemistry models became available. So, and then I talked about how long it takes for a drug to take it from discovery to commercialization, and then what are the financial aspects that are involved in that process. And then I talked about what are the main goals of the drug development process, and which is from identifying the targets and validating the targets and understand the disease, and come up with lead molecules and optimize the lead molecules to achieve desired safety, and take the lead compound to preclinical and then evaluate the safety and efficacy, and then move it to clinical studies to approval from regulatory agencies. Then I discussed about the two arms of the development process for drugs, which is discovery and development. And the discovery phase, there are two different types of, the discoveries, the structure-based and target-based. And then how the structure-based, what are the differences between the structure-based and target-based discovery. And then the development through preclinical, clinical, and post-market studies. So this slide talks about the overall overview of the target-based discovery and development. So, in this slide, I talked about what is a drug target. I think this is where I stopped, I guess. So, what is target means? So, what are the drug targets? So, the drug targets are a specific macromolecule or a biological system which drug will interact with. So these target identification sometimes can happen through incidental observations or serendipities. We have seen in research the drugs developed for one indication and accidentally they found something else. And the sildenafil is such an example. And then do basic research into the disease process and its causes and understand the biochemical and biological processes of those diseases and identify the targets where actually if we do the intervention we can bring a desired results to provide cure or improvement to a disease condition. So the target identification, there are several computational chemistry tools are now available, such as in silico approach or molecular docking and scoring procedures, proteomics and genetics. These are all the techniques that initially used by the researchers to understand and identify the right target inside our body. So I am going to give one example which I extensively worked on and which is now approved. We have a drug approved for this disorder. It's called a non-24-hour disorder. So first of all, many of you might be wondering what non-24-hour disorder is, and you may be wondering, is there a disorder or something like this even exist? Yes, it does, which is actually a chronic and very dangerous disorder. So it's actually a circadian rhythm disorder in totally blind patients with no light perception. So people who are blind with no perception, they don't know when it is dark and when it is light and when it is day and when it is night. So what is circadian rhythm? So circadian rhythm is actually a natural and internal process inside our body that regulates our sleep-wake cycles, repeats on each rotation of Earth, roughly around 24 hours. So usually every patient has their circadian rhythms are set to little over 24 hours, but a healthy subjects and the sighted people, their brain readjusts that to 24-hour clock based on their day and light cycle. So these circadian rhythms also can refer to and other biological processes, and that can display some entrainable oscillations about 24 hours. So what are entrainable oscillations? So I will talk about that in the next slide. So the 2007 Nobel Prize in Physiology went to Jeffrey Hall, Michael Robash, and Michael V. Young for their discoveries of molecular mechanisms that actually involved in circadian rhythms. So I talked about these entrainable oscillations, and the entrainable oscillations are our routine day-to-day lifetime things. So for a healthy subject, if you believe or not, everyone, for each individual, it is separate. It could be 2 a.m., 3 a.m., or something like that. But your peak sleep, deepest sleep will happen for you at the same time every day. So that's how rhythmic our body actually entrained for this 24-hour daylight cycle. So for example, 4:30 a.m. in the morning, your body temperature is the lowest. For majority of the healthy subjects who has good sleep hygiene, their sharpest rise in blood pressure happens around 6:45 a.m. And then melatonin secretion stops at 7:30 a.m. and their bowel movements starts. And then at 10:30 in the night, your bowel movements surprise, so that your body actually prepares you to go to sleep and have now other biological disturbances during your sleep cycle. So these are well entrained uh, day-to-day life events. These oscillate through the daylight cycle and in the circadian, which are called our circadian rhythm. So what is that actually controls this, to make this completely fully entrained and put it in order, there has to be some mechanism inside our body that should be controlling all this. So obviously it is our brain. So there is a central pacemaker in our brain, which is called suprachiasmatic nucleus. So this suprachiasmatic nucleus, when it sees light or when it sees dark through the retinal glands, it receives the signals and it sends the signal to pineal glands inside our brain. So that actually secretes a chemical called melatonin. So that melatonin actually causes us to go to sleep. So not just the sleep, but there are other inputs such as our social contacts, our food, and our physical exercise. These are all are certain inputs that our brain and that central pacemaker actually processes and and then uh, gives a response to other organs of the body, which are actually also entrained. So this activated suprachiasmatic nucleus actually sends signals to our neuroendocrine system, and that actually sends signals to other parts of the body. So which is all are aligned through our heart rhythms and our renal functions and our hepatic functions and the liver functions. Everything is oscillated and entrained to this 24-hour cycle. So in non-24-hour disorder, usually as I explained, sighted people that body clock is entrained to this 24-hour cycle. And for people who are totally blind, they're unable to perceive light. So the body clock and their sleep-wake cycle can cause, it can become out of sync with the 24-hour day-night cycle. After each day, their sleep cycle moves by few minutes or few seconds. And then they will have an episode of extreme sleep, deep sleep cycle, 5 a.m. in the morning, or 12 noon at the afternoon. So that actually causes, because the all other functionalities, their neuroendocrine system, their liver and heart rhythms and the renal functionality, everything is associated with the circadian rhythms. Because of this shift in their sleep cycle actually triggers other side effects blood pressure and their hepatic impairments, mood swings, and it can cause to a ton of other side diseases with this. So this is how dangerous this disease is. So, and as I explained, the melatonin secretion actually plays a major role in sleep-wake cycle. So intervention to those melatonin receptors could result in readjusting our sleep-wake cycle. So the melatonin receptors are the target. So now the target is identified. So the target is validated, and there are several melatonin receptor agonists that are like developed like tazimiltian, ramelitian, and agomelatine. And of these, tazimiltian is the first drug that actually reached all the way to the finish line, and it is now approved to treat the non-24-hour disorder in blind subjects. And the circadian rhythms, it's not only non-24-hour disorder. So this drug can also be actually it was also evaluated in sleep-related disorders for children with Smith-Magenis syndrome. Smith-Magenis syndrome is actually a genetic disorder, and kids with the disorder, they have a reversed sleep cycle. Their melatonin secretion is peak in the daytime, and the melatonin secretion is actually less in the nighttime. So they are fully active and awake during the night, and they are completely sleepy during the daytime. So to treat that, the SMS clinical trials has completed, and now the drug, we submitted a new drug application for that indication, and it is now being reviewed by the FDA. And I think December 1 is our PDUFA date, and we will hear from the FDA whether the drug will be approved for the indication or not. And we also completed clinical trials for the jet lag disorder for this, because this tazimiltian is known to entrain our biological clock. So people who has travel regularly, across the oceans, on a daily basis for businesses, this jet lag can cause and disrupt their activities post travel. So, and many may have already experienced this, who traveled overseas. So we finished a clinical trials in jet lag, and we submitted a new drug application, but the data was not sufficient, and we are in talks with the FDA to generate additional data and get the approval for jet lag disorder as well. So if approved, that would be the first medication available for just jet lag disorder. So then once I explained an example of the target identification, and then I will take you to the next step of the development process, which is the lead compound identification. So now the researchers are has plenty of information on the mechanism of the disease and the targets, they identify, and then they try to find the right molecule that could become a drug candidate, and that could become uh, um, that could intervene the biological process involved in the disease, and then bring a cure. So several sources, nature has been offering the very useful substances. For example, like penicillin, which is derived from the green molds of genus Penicillium. So the almost about 26% of the approved drugs so far right now in market that are in market are actually derived from nature. And also there are advances in the computational chemistry, actually now allows scientists to create molecules from scratch. They can use sophisticated computer modelings to predict what type of molecule may work for a given target. And the third approach is to find a lead compound is high throughput screening. So this is the most common approach majority of the pharma companies and researchers usually take to find the leads, like, using robotics and computational power. So they screen hundreds and thousands of compounds against the target and identify a few lead compounds that are that look some promising, and then based on that they synthesize a library of compounds, and then from that library, these library of compounds can be tested, and then a lead compound can be selected based on the target affinity. And also biotechnology and genetically engineered living cells, like, living systems can be used to produce some disease-fighting biological molecules, such as insulin and vaccine products, are some examples for biotechnology and macromolecule drugs. So once the lead is found, the next step is to lead optimization. So what are the steps involved in lead optimization? Lead optimization, like, is mainly to screen the identified lead for its safety and toxicity. So, and then you identify the pharmacophore, which is actually responsible for the biological activity, and keeping that pharmacophore intact, you can change the physicochemical properties of the molecules to attain certain desired results and certain safety levels. So, and the based on the affinity of the certain compound towards the target, the structure can be altered. For example, these structure alterations could actually, help a more active substance, a more active and more affinities drug, which can sometimes it can reduce, eliminate some excessive functionalities on the drug, and then reducing the toxicity, such as, nitrates or, aldehydes groups, and the halides and amines. They decide unnecessary, safety issues, and you modify the drug in a way that, you take off those excessive functional groups in the molecule, so that, you achieve a desired and comfortable safety levels. And also sometimes, you it depends on the chemistry, for example, like the target organs and the physicochemical properties of these drugs, for if you are targeting, if your target is in the brain, so you need to develop a drug that is actually, can cross the blood-brain barrier. To do so, the compound should be more lipophilic. So and also, at the same time, you don't want to make your compound, least soluble, and uh, the, the solubility of these compounds can be improved by structural alteration, reducing the more organic moiety, and, eliminating the more carbon load in the structure, can actually result in higher solubilities, and which actually, helps the drug to get absorbed and more bioavailable to the target. So, and then further lead optimizations, the once these lead compounds can be tested for, their ADME, and sometimes, the ADME is, done in the preclinical stage, and some researchers prefer to do it in early on the stage, so that, they get more understanding of these lead compounds. So these studies are conducted to test the compounds in living cells and animals for their toxicity levels, and their affinity levels to the receptors, like by doing receptor binding studies, and also most recently, there are some computational toxicology models became available. Using these tox models, one can predict the toxicity of the molecules, and what undesired side effects that drug might become. These computational models, the acceptance of these models by the regulatory agencies such as EMA or FDA, which is still in its infancy and require much more rigorous validation to become more acceptable. So the chemistry part of the lead optimization. So once the lead optimization is complete and the lead compound is identified, chemistry division is actually simultaneously work on optimizing the synthesis process, and they develop a suitable formulation, identify the correct route of administration and to get a desired absorption capability, and then improve bioavailability. So there might be some drugs which can are potent towards the target, but their solubility are much lower. So the formulation development is the stage where it is the excipients are added to improve the solubility, and wetting properties of the drug can be altered using by adding certain excipients, and then you can make more soluble and then improve the bioavailability of the drugs. And also evaluate polymorphism, and and if this uh, exhibits several crystal forms and or solvates, and then identify the most stable crystal form, and then generate impurity profiles and identify if there are any potentially genotoxic impurities that are present, and also scale up the process of the manufacture to manufacture some kilo scales required for the preclinical studies. And also another thing is qualitative and quantitative analysis of drugs. There are several analytical techniques as the HPLC, high performance liquid chromatography, and there are gas chromatography methods, and X-ray diffraction, and there are several quantitative and qualitative analytical methods, and develop these methods to um, control the quality of the drug, and validate those methods, and then monitor the stability of the active substance and also the final formulation. So after all, the once when, in order to reach the market, a drug should uh, possess a good stability in a the most desired is it is stable in room temperature. There are several drugs that are actually um, uh, they prescribe the prescribed drugs that are actually requires refrigeration and all, but the most desirable is that they are stable at room temperature. So that's the discovery part, and moving on to the second arm of the drug development phase, which is the development phase. So in the development phase, there are two, I can say three stages, which is the preclinical development and the clinical development and post-approval development. So preclinical development. So the preclinical development usually analyzes the bioactivity of the substance and the safety and efficacy of the formulated drug product. So these tests are commonly called the IND enabling studies. IND means investigational new drug applications, which is filed with the regulatory agencies to asking permission to test the certain drugs to take it to humans. So these studies are conducted to evaluate potential toxicity, evaluate pharmacodynamics and safety pharmacology, and pharmacokinetics, including how the drug is absorbed and how it is distributed in the body and how it is metabolized and excreted from the body. And also the radio labeled mass balance studies and single dose and repeated dose and reproductive toxicity and genotoxicity studies, whether the compound is genotoxic and creating any mutations to the DNA. So, and depending on the route of administration and the treatment class, some additional tox studies such as local tolerance. For example, if you are actually developing an intramuscular injection product, the local tolerance where the at the injection site, is evaluated during the preclinical stages. And going to the step-by-step of the preclinical development, pharmacodynamics. Pharmacodynamics is something what a drug can make affect your, how a drug can affect your body, what a drug does to your body. So that is the usually pharmacodynamics. It studies and evaluates the therapeutic effects of the drug. And there are several animal models that are available to test the pharmacodynamics, and also sometimes the biomarkers are available to assess the pharmacodynamics and efficacy of the drugs. The next, going to the safety pharmacology. The safety pharmacology is the studies effects of the drug on central nervous system and the respiratory system. These are conducted through repeated dose toxicity studies, they check for any abnormal and adverse events in the respiratory or CNS systems. And also they evaluate for cardiovascular safety studies using the HERG assay. HERG is the gene that codes our potassium channels, and the HERG assay actually looks for if there is any inhibition of the potassium channels, and if there is an inhibition, actually that's an indication of cardiovascular toxicity. And the next, the other part of the preclinical development is the pharmacokinetics. So pharmacokinetics is how the drug, once entered into the body, what happens to the drug. So the pharmacodynamics is what happens to the body when the drug is given, and pharmacokinetics is what happens to the drug, what the body does to the drug once it is given. So it usually typically includes the in vitro metabolism studies and the plasma protein binding studies, and as well as systemic exposure. Systemic exposure is in your blood plasma, what are the exposure levels, what are the AUCs and Cmax, and these pharmacokinetic properties are evaluated in the preclinical development. And ADME and radiolabeled mass balance studies. The radiolabeled mass balance studies, sometimes they evaluate to designate the classification BCS, biopharmaceutics classification. And also they use C14 and tritium labeled drugs to conduct these ADME and radiolabeled mass balance study. The labeled drug is given to the body, and then using LC mass spectroscopy, they identify and quantify the metabolites and then measure the mass balance by sacrificing the animals, and how the drug is distributed in different parts of the body, and what are the major metabolites expressed in the urine, and what are the major metabolites expressed in the plasma and feces. And this comprehensive understanding of how the drug is absorbed, distributed, metabolized, and excreted from the body. And then comes to the main toxicology studies. The one type of toxicology studies is acute studies. The goal of the acute study is to determine toxic dose levels and what is a lethal dose, and what level of dose is needed to be lethal, and then what's the maximum tolerated dose, and also what's the dose where the no observed adverse effects are observed, which is called NOEL. So these parameters are important for predicting the human safety and the dose selection for the clinical studies. And in the repeat dose tox studies, the duration of these repeat dose tox studies depends on the duration of the study and the types of diseases and acute or chronic, depending on that, the design of the repeat dose tox studies are determined, and the dose levels and the dose regimens should be selected so that the observed exposure of the drug in the non-clinical species is adequately covered for the clinical exposures, which is the human exposures. You take such a large concentrations in animals so that you can have an estimation of these levels, what are the safe and levels to start for a human clinical trials. So for all these IND enabling studies needs to be done at least 2 mammal species. And then the genotoxicity. The genotoxicity is the gold standard is the AIMS study, which is reverse mutagenesis study that is evaluated if the compound results any undesired if the compound is mutagenic and cancer causing. And also reproductive tox studies are needed before a drug can be administered for women of childbearing age. There are 3 segments in this reproductive tox study, which are the segment 1, actually tox studies look at the effects of the drug on the fertility levels, and the segment 2 and 3 that actually looks postnatal development and endocrinic levels, what are the effects on the endocrinic system, and also postnatal development of the uh, the embryos. So though based on these preclinical data and a safe clinical dose to start with in humans is determined, and a clinical study protocol is developed. So, and then before going into humans, you have to submit a new drug application to the regulatory agencies. So these IND filings usually involves the preclinical toxicology study data and your CMC information, which is chemistry, manufacturing, and controls information, and a clinical trial protocol. So the CMC typically involves the quality information of the active substances and if the impurity levels are adequately qualified using the tox studies, and also the quality of the final formulated drug product and the stability and how stable is the formulated drug product. And the clinical trial protocol usually typically involves the clinical trial plan, and that outlines how the study is conducted and where and what are the what population it is being conducted and where and who will be conducting these studies. So apart from the IND, usually human clinical trials also require the approval from the local institution review board, which are usually called IRB, and also ethical committees. You need the approval for the ethical committees. So moving on to the clinical development from preclinical to clinical stage. One is the phase 1 clinical studies. So the phase 1 clinical studies, the drug candidate is actually usually tested for the people for the first time. And these studies typically involves 20 to 100 healthy volunteers. And the main goal here is to evaluate this safety, and then using the dose escalation studies, what are the safety doses, what's the maximum efficacy dose, and what's the maximum tolerated dose in humans, And the phase 1 studies look for all those, like, and then pharmacodynamics and pharmacokinetics are also evaluated during the phase 1. And these closely monitored trials are designed to help determine, the safe development of these drugs. And then at later stages, like, when we move to the stage 2 or phase 2 or phase 3, some additional phase 1 studies are also, like, conducted for, such as, like, drug-drug interaction studies and bioavailability in hepatic impaired, like, or renal impaired people, and the effect of smoking and effect of alcohols, like, how the smoking and alcohol affects the bioavailability and absorption of the drugs. These phase 1 studies are conducted much later into the development stages, such as, like, mostly in parallel with phase 3 trials. So overall, phase 1 actually, like, helps to develop a comprehensive, prescription label information, and, like, and design an appropriate phase 2 clinical trial. So in the phase 2 clinical trials, like, the drug is evaluated on a little bit more larger scale in the disease condition, whereas phase 1 is actually in healthy volunteers. In phase 2 is actually mostly proof of concept studies, and that are conducted in the diseased patients which has the disease, and they assess the possible short-term side effects and the risk associated with the drug and how they are responding to the drug, and the efficacy and safety of these drugs are actually usually evaluated, and, like, is it the drug working by the expected mechanism, or is it improving the conditions? And also, like, during this stage, like, they optimize the appropriate doses, and, like, whether it's a once a day, or, like, based on the pharmacokinetic profiles and absorption and bioavailability and the Tmax, which is actually, like, the time that is required to achieve maximum concentrations in our systemic, which is in plasma. Based on all those facts, like, they determine, like, the duration of the medication, and how often you need to take the medication, once a day or twice a day. Those type of, like, a more thorough evaluation is done in phase 1 and phase 2 studies. So if the drug continues to show promise in phase 2, and then they prepare for the much larger phase, which is the phase 3 clinical trials. Before I move on to phase 3 clinical trials, I would like to, like, cover some chemistry related aspects for the phase 1 and 2. During this process, like, you further improvise your synthesis process to improve the yields and establish a better impurity profiles. And also, like, these days green chemistry is something that is becoming more popular and popular. So these solvents, the choice of solvents and the levels of solvents that you choose should not result in a huge environmental impact, and you choose safe solvents and minimum volumes. These are all the aspects that are actually, like, considered in this phase chemistry development. And then scale up, obviously, like, to multi-kilo levels to evaluate appropriate GMP starting materials, and the GMP procedures are established. And then coming to the formulation and the drug product, based on the initial results, the dosage form, then the dosage strength, and the composition may require further optimization of the final formulation to improve the dissolution or bioavailability of these drugs. And for example, if it is actually a pediatric drug, like, you do the test masking, and organoleptic properties are evaluated so that the children who take these medications actually, like, they which is well accepted by the children, for it is the drug is indicated for pediatric population. And then you evaluate the photostability and stress stability conditions of the drug to better understand the degradation pathways of the drug, and then so that over the period of the time, what type of impurities you might be expecting on storage of the drug, and then validate all these qualitative and quantitative methods. And the another thing is the conduct the container closure integrity test. When if a liquid drug is stored in a container, you should evaluate for the extractables and leachable, and you should make sure that you choose containers that doesn't leach any resins into your product. So going to the phase 3 trial, this is the final stage of the drug development process. In phase 3 trials, you go on test your drug on a larger set of patients, like maybe 1,000 to 5,000, and sometimes several 10,000s of patients to generate statistically significant data to show that your medicine is effective and your medicine is safe. And it gives a risk benefit relationship, and they evaluate the overall risk benefit relationship of the drug. So because we are running out of time, I just would like to wrap up quickly. And phase 3 CMC considerations, mostly the chemistry considerations on the active substance is establish the critical process parameters and develop a robust and reproducible process that can give you a consistent quality and yields, and then validate that manufacturing process and identify and designate a GMP starting material and establish some quality requirements, which we call as specifications for the starting materials and reagents and final drug, active drug substance. And the complete characterization of impurities and their control strategy, how you purge out those impurities from your final product, so that your final product is more pure, and at least establish 1 year of long-term stability upon storage of these batches on long-term conditions, and provide justification of your quality attributes. For example, if you are designating a certain purity numbers or 98% or 99%, or if you are providing a specification, a certain impurity should not be more than this. So you need to justify why you think those levels are safe in your final substance, and also develop reference standards for the regular analysis of the drugs. And coming to the development aspects of the formulated final drug product, excipient compatibility, you add excipients to the active moiety to make the final drug product, and then you need to check the compatibility of those excipients and compatibility of the container closures and diluents. For example, there are some medicines which are supplied as solids, and then you use a certain diluent aqueous or diluents to dilute the drug to before you administer. So if you are using diluents, compatibility with those diluents, and the in-use stability studies and freeze thaw stability studies, and these are all the aspects that you study during the phase 3 of the final drug product development. And another aspect is biopharmaceutics evaluation, the dissolution, in vitro and in vivo correlation, how the drug is dissolved and absorbed in the body, and what kind of modifications that you can do to achieve absorption, and sometimes the there might be adverse gastric effects in your gastrointestinal systems, to mitigate those risks, what are the strategies and how you actually modify your formulation. For example, aspirin has known for gastric side effects, and now that they coat the aspirin with enterin, so that it is not absorbed in your gastric in stomach, it is the enterin coat actually protects, and then the most of the absorption of the aspirin is actually as is done at in DODM. So such are the aspects like you develop in the phase 2 and 3 stages, and the BCF classification and the bio waiver is actually when you are applying for NDA approval. So some the waiver to for bioequivalency studies may be and bioavailability, absolute bioavailability studies may be waived based on the classification of the drug. And there are 4 different classes for this, high solubility, high permeability, and low solubility, high permeability. Based on this, there are 4 different classes. Sometimes even if you meet criteria using the BCF classification system, FDA might look your the radio labeled mass balance studies, based on that, for example, if it is a high solubility, high permeability compound, they don't just designate class 1 because if your mass balance is not achieved in the radio label mass balance study, the recovery is less than 80%, they usually designate class 2, they don't consider as class 1. So these are the aspects that are involved in the CMC development of phase 2 and 3. And also finally in the phase 3, the critical process parameters, if it is a capsule or tablet or a suspension, a sterile injection, what are the critical processing parameters for that manufacturing? Those are identified, and then the process is validated. And finally, one other aspect is the environmental impact factor of the drugs. Once the drug is excreted from your body and the wastage of these drugs, and most of these drugs, some people dump in these trashes, and how this drug is accumulated in the through our drains, it might go to the river, and the how the sedimentation on the in the river systems, and those aspects also needs to be evaluated before you take it to the final approval. And an environmental impact factor is calculated based on how much of the drug you are going to produce in first few years of your commercialization. And then if the environmental impact factor is below certain levels, you can ask the regulatory agents for categorical exclusions, or if it is above those levels, you need to conduct environmental toxicology studies for as aquatic stocks studies, how the aquatic influenced by the trace levels of these drugs in our river systems, sewage systems. And then once you have all this data, you submit for a new drug application to the FDA, and they review the data. And sometimes if it is a new class of diseases, they also appoint a advisory committee. The advisory committee consists of the top researchers and clinicians in that disease class areas. They meet with the FDA and they evaluate the data and they give an opinion of the drug. And the final basis for the approval is always the risk benefit ratio. So, and what are the risks involved with the drug? Any drug, no drug is actually foolproof. So there are always some side effects with every drug that is on the market. So based on the benefits that a patient or people receive from the drug and what are the risks associated with those, based on that risk assessment, risk benefit ratios, FDA actually usually decides whether it can be approvable, or they can send sometimes an approvable letter requesting more information and more studies, or they can simply deny the approval saying that the risk is more compared to the benefits of these drugs. And the final is the post-approval stage. The FDA asks companies to monitor the safety of the drug, post-approval. And they sometimes they also require us to do some post-approval studies, which can be conducted after the drug entered into the market. And after that, you do um, the you report the periodic safety updates to the FDA and your annual reports, um, the in a particular year how much the drug is distributed and what was there any other adverse effects, and like all the adverse events in the entire patient population needs to be completely compliant, and periodic that safety updates needs to be submitted to the FDA. These are all the post-approval activities. I just running through because we are already ran out of time, and I am open for any questions, and would like to thank the university for giving me this opportunity.
Thank you, sir. Thank you for your wonderful lecture on drug development, discovery to commercialization. Now, I request to participants, if you have any doubts, the session is open for discussions. If you have any doubts, you can ask. So you can see in the comment box, excellent presentation. Thank you for your presentation, sir. And all the participants, if you have any doubts.
I think there are no doubts because
Yes,
you
yes.
left no doubts, Ravi, because you have covered everything. It is elaborate at the same time very lucid. Extensively, you have given every detail of it. So that's the reason why I think the students are not having any doubts.
Thank you so much.
Absolutely no. Absolutely no. You have covered everything, each and every inch of it. In fact,
Thank you.
detailed, yeah, nice detailed drug development from Shanta.
Madam, all the topics, sir, which discussed, those
Yeah,
are
they
included in our
syllabus.
fourth paper, first
Fourth
paper.
paper. The thing is, Ravi, I never taught drugs.
Morning session students wrote exam.
The irony here is I never taught drugs for MSc PG students.
I think even students got the question, clinical trials. What are clinical trials, and write a brief note on clinical trials.
We can
Same
give
question
it.
they had in the morning session.
Morning session. Okay.
Okay. Sure. I can share my slides, madam. If you want to, forward this to students.
Yeah, definitely. You please do that. You mail to my. I have given the mail ID, no, isn't it?
Yes, ma'am. I do have your mail.
Really excellent, and you have given without any break in between continuously. I think for more than an hour you have given elaborate, extensive, and still it is very clear even to the student who attends for the first time. He will attend right from how what is meant by a drug and how it is commercialized, how it is discovered, and how it is commercialized. So
Thank you so
excellent.
much.
So before actually I give the concluding remarks, I just wanted to say a few words about Ravi Kumar, and I'm really very, very proud to claim that he is my student. Actually, in fact, he was a student of AV College for his post graduation during the year 95, 97, I think, if I'm not wrong, Ravi.
96, 98.
96, 98, 96, 98 batch when I was the faculty member there. So I'm really proud to say that he's my student, and this again shows that um, sincerity, commitment, and hard work has no boundaries. So he has started his journey from Mehboob Nagar, but now he's placed in such a high reputed institute where he is, in fact, is saving so many lives. We can say not exactly saving, but he's into the healthcare system. So definitely, if you put hard work definitely you can reach up to any level. And this is especially to my students, Palamuru University, where this university is established in a very remote area. Generally, students will be under the impression that it is very difficult for us to reach up to such levels. And most of the students, even after pursuing MSc chemistry, and I always say that, see, the students who are coming into MSc chemistry course, especially the PG MSc chemistry course, they should not stop their studies at MSc level. And most of the students here, generally, they are happy or they're content with petty small jobs, and not, I don't have any disrespect for any kind of a job, but generally the students who are opting for MSc chemistry, they should not stop. My intention is that they should not stop at the PG level. They'll have to pursue higher studies. They have to definitely go into research, and so definitely we have lots of responsibility as chemists. We have lots of responsibility, as just now he said, Ravi. He said that we are really at advantageous stage where we are not only involved in the synthesis of the drug but also in the process development. So how he has explained clearly. See, normally we think that we are only involved in the what the synthesis of organic molecules, which can be used in healthcare systems. But he rightly pointed out that point. So where it, the chemist has, they have a dual role. Anyway, we can use our talents. So at the same time, today coming to the topic, he has covered every point of it, where the drug development, in his words only, I'll say, just two arms. One is a discovery arm, the other is commercialization. And he actually covered every point in discovery arm, where it is first how it is. The target is based on the structure, and then target based, and then identifying the lead molecule, and how you are going to identify what is a pharmacophore, and then how you are going to develop before. And after that, coming to the commercialization process, it has to undergo, before a drug is marketed in as a safe drug in the market, definitely lots of pains are involved. It has to undergo many stages, that is preclinical, clinical, and the approval stages. And definitely it takes not just 1 or 2 years also. Sometimes it may take years to together, that is 10 to 15 years also may take. And lots of money is spent on this. And after doing all this, definitely we'll have to see to it how the drug is very safe to the patient, and then taking into consideration all the effects, like it should be at the same time environmentally, it should not affect the environment, and then all the aspects. And finally, what is the amount of money spent on it, and finally when it is coming to the commercial, when it is coming to commercialization, definitely how viable it is to the companies who are actually involved in this process. So considering all these factors, this is what is the drug development. And I'm happy that he is involved in the one of the drugs, that is sleep disorder, that is circadian rhythm. He talked about circadian rhythm, and when a circadian rhythm is disturbed, how it affects not only the sleep but the other biological process in the body. So definitely this is the problem to be addressed, not only in the blind people, because of the light perception. Definitely they'll have this disturbance, and the drug is developed is Heltios. Is it Heltios?
Yes.
The drug, Heltios. And
The active drug name is tasimelteon.
Yeah, tasimelteon, and it is a melatonin receptor. So at that junction, he talked about the receptor, that is how to identify the target. So definitely, and it is not only used there, and later on it is also applied for children suffering from some sleep disorder disease, that is Smith Magonis, where generally student they will have what a high sleep melatonin secretion at the peak level in the reverse order. Like normally you should have in the nights, and less in the daytime, but it is reverse in some children. And so there also it is addressed. And not only that, jet lag people, like generally we move, most of the people for business, they move across the oceans, and for them definitely jet lag is a problem. So even there it is addressed, this particular drug. And it is already in the clinical, it is all, it has, I think it is marketed, isn't it? It's been
Yeah, it is
used.
marketed for
Yeah.
the blind indication,
Blind,
madam.
now it is testing for other jet lag and children, isn't it?
Yeah, the testing is completed and it is with the FDA
Phase
for
3,
review, madam.
phase 3 stage it seems. Is it
No, phase 3 is also completed, madam.
completed?
Phase,
Okay.
yeah.
Okay.
Now FDA is actually reviewing the data whether it
Yeah.
can be approved.
So nice, and I try to jot down the points because for the first time I'm listening to all this, but I could get only some of them. But still, the students, because I think students don't need any information right now. Like I'm no concluding information is required for them. I believe because you have given so clearly, you have covered each and every point, and I think the students are greatly benefited. I'm once again very happy. If I say thank you, definitely Ravi feels bad being my student, and so I'm saying definitely the students are benefited.
Sure,
So
thank you,
good,
madam. Thank you so much
good
for
Ravi.
inviting. Yes,
So
yes.
after a very long time, see after your MSc, I think I'm seeing you. I don't
Yes,
exactly remember your face also, but I could make out that. Yeah, vaguely I remember, but still, I'm very happy to see you again. Very recently, we happened to meet.
Yes, madam. Yeah.
Yeah, but I think 1 month ago, and
Yeah,
today
1 month ago, actually,
yeah,
Vivek, I think Vivek informed me about this,
yeah.
and also Dr. Ramudu also contacted
Vanaparthi, yeah.
to participate in your
Yeah,
facilitation
yeah.
thing. And
And one more thing, students, especially for the students, I'm telling this is what is a student-teacher relationship. Students, this you'll have to remember. You should not forget your the origin. See, he when immediately when I asked him, see the other day when we met in a felicitation program, I just asked him, Ravi, like, can you please give any lecture, or are you like, can we make use of any services? Then immediately accepted it. So even after a very long time, because in spite of his busy schedule, he immediately made it a point, because I have asked him, he made it a point to, even though today is a working day for them, he made it a point to give a lecture just because I asked him. So I'm very happy. So this is what you'll have to develop, not only the studies part of it, even this you'll have to learn from such students. Okay.
thank you so much, your kind words,
Thank you,
madam.
Ravi, again.
Thank you.
Nice. Sudarshan?
Now I request Mr. Sudarshan Reddy to present a certificate of appreciation to the speaker. Thank you, sir, for being with us and
Thank you, madam.
extending.
It's always pleasure to be part of and anything that I could help. When I visit to India next time,
India.
like, definitely will make sure to connect with madam and yourself and see, like, come and visit the department.
University. University.
University.
Yeah,
Come and
definitely,
visit the
sir.
department.
Yes.
For you, sir. And really, as Madam said, it's a student should inspire by seeing you, because you are also originated from Mahbub Nagar and you reach a very highest position in the field of chemistry. So I think it's really inspirational to the students, and they can be motivated by such interactions with eminent speakers, or subject wise also, as well as personality wise
Yeah,
also. So
personality.
Yes.
it's a really a good opportunity to the students to interact on this stage, on this platform. So we are making use of online technology for filling the gaps in this COVID situation, because students are not reaching the universities physically, and we are trying to fill those gaps, and we are regularly making them some topics or some interactions in the field of chemistry, so that they will be in a continuous phase of learning the things. So I extend my gratitude and thanks to you once again, sir, for being with us.
Yeah, thank you so much, madam, and thanks for your words. And also for students, I would like to say one thing is that apart from the hard work and dedication, one other aspect is the passion.
Yeah.
And have passion towards whatever that you do. I read a book about passion. It's called The Fred Factor. A postman who delivers a
Yeah, I've
postman.
seen. Yeah.
So
Winner.
the passion about his own job, it's a
Yeah.
simple job, but like no
Yeah.
passion in that how he makes a difference in the
Yes.
community.
Yes.
And
Very
whatever
good.
that you do, if you have passion and you can reach highs, that's
Yes.
all I can say to the students. And build interest, build passion towards what you do. Thank you so much, madam, for giving this opportunity.
I think that's good. Good, Ravi. Very happy.
Thank you. So now we move on to the next speaker with us, and we are coming to the last speaker, Dr. Hari Krishna Reddy Garu. Uh, Hari Krishna Reddy Garu.
Good evening.
Yes, sir. Welcome to the virtual international webinar, sir. And I request Mr. Gnaneshwar to introduce the speaker to the audience.
Thank you, madam, for giving me this opportunity. And good evening to one and all. On behalf of Department of Chemistry, Palamuru University, it's my great pleasure and an honor to introduce the eminent personality, Dr. Hari Krishna Reddy sir, Senior Principal Advisor, Novartis Institutes for Biomedical Research, United States of America. Sir, master degree from University of Hyderabad and MPhil from University of Hyderabad, doctoral degree from University of Hyderabad under the able guidance of Professor Devi Basavaya sir. Sir has published more than 40 research papers in various research areas in a reputed national and international journals. Sir has worked as a co-inventor on 10 patents related to cancer field. Sir has got Young Scientist Award in 1993, organized by AP Academy of Sciences. Sir has worked as a research associate in various national and international universities. Sir has a lot of professional experience in various pharmaceuticals as scientist, research advisor, senior research advisor, and senior principal advisor from 2001 to till date. Now I welcome Dr. Hari Krishna sir to deliver a lecture, and over to Hari Krishna sir.
Thank you, Mr. Gnaneshwar. Thank you for your nice introduction. There is a small correction. I did my PhD with Professor Govardhan Mehta, and I did my MPhil with Dr. Devi Basavaya. Okay, with that, I will share my screen. Please I will make sure. Oh, Mr. Ganeshwar, it says host disabled participant screen sharing. Can you please able? It says disable the screen sharing.
Screen sharing disabled.
Yes, sir.
Is it fine now?
Yeah, you can share now.
Yes. Yeah, I got it. Okay. It's my honor and privilege to present my presentation at the Palamuru University in multiple reasons. One is, of course, Palamuru University is a district level headquarters university, which is also my hometown. I am a product of the Palamuru University, citizen of the Palamuru University, and I studied also my intermediate in government junior college and my uh, degree BSc in MVS Arts and Science College. And today I'm going to present significance of organic chemistry in medicine. And this afternoon speakers, Mr. Ramu and Ramu presented a total transition metal chemistry, how one can do the methodologies and different arene systems. And Dr. Ravi presented how the drug discovery system goes on from the discovery to the manufacturing and market stage. But here now I will only focus on the discovery chemistry level, and also I will not go in detail of the complete total synthesis, but I will give the glimpses of um, synthesis, how organic chemistry and organic chemist can achieve and synthesize complex molecules. And to begin with, in the benefit of students, I will start with the fundamental organic chemistry. And organic chemistry, as you all know, is a chemistry of carbon. And before that, I will just show the same flow chart. I think this flow chart basically Dr. Ravi Kumar presented from organic and medicinal chemistry from the discovery to medicine level, how it goes. But first, organic chemist in the discovery level synthesize the molecules, and then the biochemistry and biology will be explored, and then pharmacology studies to show all the pharmacokinetics, and then toxicology studies, pathology studies, and finally, after all, the clinical trials, internal medicine. This is just to show flowchart how the organic chemistry can go into the medicine. And first, start with carbon, because this organic chemistry starts with carbon. And as you all know, carbon, is a four valency system with the dihedral angle of 109.5 degrees. But however, the chemists can make some of the compounds varies from this dihedral angle. So you will know by end of my talk, you will see how that will be affected. And also the simple carbon and with inorganic chemists ants, how complex molecules can be synthesized, and what is the power of organic chemist. So, okay, just to simple representation of carbon, it has three hybridizations, sp3, sp2, and sp hybridization. And depending on hybridization, you can form the alkanes, alkenes, and alkynes. And this is just representation of the molecular models of methane, is a tetrahedral, it's in the ball and stick model, and space filling model, and etc. And if you go back to the hybridizations, and of course sp3 hybridization will form alkanes, and all sp2 hybridization forms alkenes with the CC double bonds, and sp hybridization forms alkynes with CC triple bonds. And another sp2 hybridization examples of arenes have a special bonds that are represented as alternative single and double bonds, which can conjugate and give more stable aromatic systems. And these are the just brief introduction of different functional groups on carbon. We can put carbon with one hydrogen, and carbonyl group forms aldehyde.
Hari,
And
Hari,
yeah, yeah,
sorry to interrupt you.
yeah.
Are you sharing these slides?
Yes, I already shared. Have you
Nothing
not
is
seen?
visible. No.
Oh, okay. Let me see what is going on. Can you see my screen at all?
Yeah, we can see your video, but not the slides.
Okay. Um, can, okay. 1 second. Can you see any of the my computer screen right now?
Yes, sir.
Girija,
Yes, sir.
can you
Yeah,
can you see
yeah,
now?
yeah, yeah.
Oh,
Now we can see.
okay. Sorry, I think some computer glitch. I guess I
Okay.
will go back and start again. I
Yeah,
think I will go back quickly.
yeah.
Okay, I think first few things are not needed because it's
Yeah.
not in the slide, and
Mm.
this is the flow chart. I just a second, one more second, so that it will be better to show. Okay, okay. This is the flow chart, just organic chemistry from the discovery to development level, which Ravi Kumar narrated all these details, and I'll start because organic chemistry starts with carbon. So I'm showing the as you all know, carbon is just 4 valency with all these physical properties with diagonal angle of 109.5 degrees. But chemists can make some of the molecules with with different diagonal angle with lower or higher depending on the strain involved, and that will come with the polycyclic compounds, and then carbon will have 3 different hybridization systems, sp3, sp2, sp, all you know. And they based on the hybridization systems, it will form alkanes, alkenes, and alkynes, and so on. And this is the one such example of simple methane molecule with the ball and stick model and space filling and other models. And based on the hybridization, sp3 will form the alkanes, but sp2 will form like a CC double bonds, which is alkanes. sp3 is the CC triple bonds, is alkyne, and then sp2 also can have alternative single and double bonds, making the cyclic more stable conjugated system, which is aromatic compounds. And with the different atoms, carbon can form with one hydrogen bond, and carbonyl makes aldehyde, and two CC bonds with carbonyls make the ketone, and carboxylic acid, esters, amides, and acid chlorides, so on and so forth. And just to show some more details of kind of organic reactions, and there are addition reactions, two molecules combine each other, elimination reactions, one molecule splits into two, or substitution reactions are part of two molecules will exchange and substitute, the substitution happens. And there is another type of reactions are rearrangement reactions. A molecule undergoes changes in the way its atoms are connected. So these are the just simple brief introduction of the reactions. I'm sure everybody knows it, but I just wanted to start with the big fundamental level and how organic reaction occurs, mechanisms. If you see the clock hands will be moving, but that is only what we see. But the mechanism behind the face is what causes the movement. So same way, in an organic reaction, we see the transformation that has occurred, but the mechanism describes the steps behind the changes that we can observe. Reactions occur in defined steps that lead from the reactant to product. So here are some simple examples of addition reactions with alkene. Sorry, alkene. You can add a water molecule across the alkene to give alcohol, and we can reduce the alkene to alkane, and we can do this hydroxylations to diol, and we can convert to ketone, cyclopropanes, halides, dialides, and halohydrins, so on and so forth. This is just show the simple reactions of addition reactions. And means I will not go through all the reactions, but there is another main important factor is stereochemistry of the organic molecules. And some objects are not the same as their mirror images. Technically, they have no plane of symmetry. And a right-handed glove is different than a left-handed glove. So the property of commonly called handedness in the stereochemistry. So organic molecules have different handedness results from the substitution patterns on only on sp3 hybridized carbon. It cannot happen on the sp2 or sp. Only certain special circumstances sp2 hybridized atom can be have stereochemistries, but that is different. And enantiomers. This, these molecules exist these three-dimensional objects. Some molecules are the same as their mirror image, but some molecules are different than their mirror image. So the molecules which are different than their mirror image are called enantiomers. Okay, after seeing all this is just introduction. Now I wanted to go into the um, chemistry, but today I'm not going to present in detailed reactions and schemes of the chemistry. I will just give a glimpses of what organic chemist can do and synthesize the molecules. And now I will go to my graduate work at University of Hyderabad, and under supervision of Professor Govardhan Mehta. I finished my PhD in 1994. Today I will not go full details of my PhD thesis, but I will take one important molecule which I synthesized, and I will narrate briefly. So here are the things I was talking about, how the dihedral angle of carbon can be changed to synthesize these strained polycyclic compounds. And I know these are like, especially this three-membered prismane called 3-prismane has a dihedral angle is the inside one is closer to 60 degrees. So, and of course, in the higher the prismanes, it will be a little bit better dihedral angle. But since whenever you want to change the dihedral angle of carbon, the synthesis becomes challenging. And the top row are synthesized in the literature before even 1994. And the bottom row ones are, some of them are synthesized by my previous colleagues, seniors in the lab. And of course, everybody knows Buckminster Fuller in C60, even much more complex polycyclic hydrocarbon. So, well, all these are whether known in the literature or synthesized by some of my previous colleagues. And in 1993, I synthesized a molecule is a C20H24 hydrocarbon with a 4-fold of symmetry. And we named this compound as a gold condane, but I will come to that part little later. And this molecule can be synthesized in about 5 to 6 steps from this starting material. And this bis norborino benzoquinone also synthesized in the lab, but I will not go into details. And if you take this compound with the cyclobutadiene, and it will have 4+2 Diels-Alder reaction, and it goes into the 2 different phases. One can add from top, one can add from bottom. And these 2 adducts were separated and did the 2+2 photochemical reaction to get this caged molecule. But if you see these molecules, I'm showing this cyclobutene red because basis showing that this top portion is coming from cyclobutadiene, and this blue portion is coming from the bis norborino benzoquinone. So once you have these molecules, and there is a difference between these two because of the facial addition of this cyclobutadiene, I will take this particular isomer, and if you see these two, because of the strain, these 2 bonds makes these 2 double bonds like a far away, like, it's like a airplane wings. And if you cleave this CC double bond between the 1,4 to the ketones, the strain will release. So for that, we can do the zinc acetic acid reduction to cleave the CC bond here. So once one CC bond is cleaved, the second bond cleavage is much more stronger conditions needed. So you have to use zinc ethanol reflux conditions to cleave these bonds. So once you cleave these two double bonds comes closer, and you can do the photochemical so 2+2 cycloaddition to get this nonacyclic compound. And in these, two ketones were reduced using the Wolff-Kishner reduction and get this hydrocarbon. This hydrocarbon can be written as like a different orientation. You can write this nonocyclic compound. Basically, it's a 4-member ring attached to the 2 5-member rings, and 2 of these are combined together. So if you see this molecule, even though it's a C20, it's a highly symmetrical compound. In NMR spectroscopy, you will see only 3 signals here. These 3 are repetitive, and all 8 centers are similar. So you will see the 3 signals in proton NMR and 3 signals in the carbon-13 NMR. Having synthesized these fancy molecules, we debated to name this compound. Initially, we were kind of thinking to name like a person's names like me or my professor, but at the end we decided to name in recognition of the city where we worked, which is Hyderabad. So it happened that particular decade was the 400th anniversary of the city Hyderabad. So since Hyderabad, in Hyderabad, one of the many landmarks are Golconda, which is the old name of Hyderabad as well. So we decided, you can see this reference of Hyderabad city in this Oxford dictionary, and in honor of the Hyderabad city, and we named this molecule as Golconda, and it has been accepted internationally. And with that, I will go further. And of course, you must be thinking, my topic today is the significance of organic chemistry in medicine. But this Reddy is telling all these polycyclic compounds. So where is the medicine? So since I worked in PhD in University of Hyderabad, I have to show what I have done in Hyderabad. So that is the reason, and of course, it is that is my foundation there. The work I have done under Professor Govardhan Mehta's lab is my foundation. All what I am today is a result of working there in University of Hyderabad. So the molecule I synthesized, as of that stage, it's not directly any use of the society, but in the literature, these polycyclic hydrocarbons, if you substitute with polynitro compounds, they act as a propellants, propellants, which can be used in defense. So you all know trinitrobenzene, trinitrotoluene, and trinitrophenol are propellants. And in previous literature, these cubane derivatives of octanitrocubane and heptanitrocubane already shown they are very high explosives, and they were using in some of the defense labs. And my word is here, that Golconda which I made. If you make, I showed just only 3 scenarios, but it can be multiple scenarios: tetranitro, octanitro, dodecanitro. If one can synthesize these molecules, they can be really act as a nice propellants. That I left it for future scientists to make it. So I finished my PhD and went further. So now I will go into my subject matter of this today's seminar, organic molecules, medicinal importance. So I will not go, unlike Ravi Kumar went from the discovery to development into the market, since I am discovery chemist. So I will focus on the discovery chemistry, and I will give glimpses of several molecules we synthesized in the lab. I will not go in detail synthesis, but I will try to give brief uh, idea of what kind of molecules one can make, how organic chemist can go what extent and synthesize these molecules. Just before going into that, I wanted to say, organic chemistry is everywhere. Organic molecules, everyday life, what you witness is the organic chemistry and organic molecules. So I am sure everybody knows and everybody needs rice every day. So main ingredient on the rice is a starch. So this amylose and amylopectin is the polysaccharides, and this is organic molecule. And if you go to the lentils, main ingredients are carbohydrates, protein, and fiber. They're all constitutes of organic moieties. Milk, main ingredients are lipids, proteins, and lactose. What are these? These are all constitution of organic molecules. This is the just everyday living. Even if I can go further, even the furniture and everything is a uh, polymers of organic molecules. So, but I will not go into those details. But I will go into the now some more common organic foods like a coffee. One of the ingredients is caffeine, which is the structure is depicted here. And there are several ingredients in the coffee, but I will not go in those details. In the tea, out of several ingredients, one of the ingredients is tannins. This is also organics. So what are tannins? Tea and coffee contains tannins, which are naturally occurring organic substances known as polyphenols. So this basically, these are the polyphenolic moieties. Polyphenols are present in other products too, like wine, beer, fruit juices, berries, pomegranates, nuts, smoked foods, legumes, some herbs and spices as well. So these polyphenols have so many medical properties, like health benefits. Polyphenols will prevent degenerative diseases such as cancer and cardiovascular disease as well. And taking these means, it will help for the sake of keeping health. So, of course, if you take the meat, also in the main ingredients are proteins, which are long chain amino acids, once again, organics. So now I will go into the medicines, common medicines. What we use is organic ingredients. It's organics, like a regular fever, paracetamol, acetaminophenone, Tylenol is organic. It's a acetaminophenone. For diabetic, metformin hydrochloride. I am not going to tell, there are so many medicines for diabetic, but I am just taking one example. So is there once again organics. So aspirin, which is very commonly used for minor aches, pains, fever, anti-inflammatory, and also used for blood thinner. So these are all organics. I just want to show how organic chemistry is used in the life. And some more for cholesterol, use Lipitor, which is atorvastatin. Calcium is also shown here. It's a little bit complex molecule than the previous ones. High blood pressure, losartan, potassium, also it's organic. And one thing I wanted to highlight here, all these medicines, of course, cures the corresponding disease or whatever, fever, cholesterol, blood pressure. However, all these molecules have a side effects.
Your voice is mute.
How about that? Now, can you see my slides? Something
Yeah,
happened
we
here?
can.
Huh? Yeah.
Yeah, we can.
Hello? Hello?
Again, your voice is mute.
How about now?
Yeah. Now it is okay.
Okay. So continuing the side effects of Lipitor, also, muscle pains, kidney problems, yellowing eyes and skin, dark urine, etc. These going forward, losartan also have side effects like fainting, high potassium blood levels, rash, itching, severe dizziness, trouble breathing. Why I'm showing these side effects is, can we, if we go further on the health issues like cancer and all those things, these side effects becomes even more uh, more devastating, that's severe. So can we do something on those side effects? That's the reason I am showing these side effects. So now I will turn into my topic into the cancer in today's world, how cancer is influencing in today's world. So if you see the first fundamental is, what is cancer? A cancer is a disease caused by an uncontrolled division of abnormal cells. So it's basically malignant growth of tumor resulting from the division of abnormal cells. So this is a practice of phenomenon perceived to be evil or destructive and hard to contain or eradicate. So these, cancer can be, uh, called as different names, like it's a malignant growth, cancerous growth, and this tumor is called malignancy, carcinoma, sarcoma, melanoma, whatever we call it. It's all kind of different cancers. And mm, see, just to give the little bit brief uh, uh, brief survey of how many cancer patients patients and cancer is affecting the whole world. See, cancer is a leading cause of deaths worldwide, accounting for an estimated 9.6 millions based on 2018 analysis year. And the most cancers are lung cancer, breast cancer, colorectal, prostate, skin cancer, stomach cancers. And the most common causes of cancer deaths are like lung, colorectal, stomach, liver cancer, breast cancers. So, and if you go further details, this is the projected new cancer cases and deaths in for a decade, 2010 to 20. So this graph shows like 1.5 million per year in 2010 to increase to 1.9 million in 2020. But this increase also, through different cancers. And of course, this graph shows the number of cancer deaths expected to increase from 575,000 in 2010 to 630,000 in 2020. However, the rate of people who died from cancer also, is more, but the number of cases are increased as well. So that is the reason there is more deaths. So what causes cancer? There are several unknowns. It can cause cancer, but some of them we know, and we can stop that. One is the tobacco use is the main cause of about for 22% of cancer deaths. So another one is 10% is like due to obesity, poor diet, lack of physical activity, or excessive drinking of alcohols. This can be avoided. Other factors includes infection to special radiations or environmental pollutions. And in the in the developing worlds, 15% of cancers are due to these viruses, Helicobacter pylori, hepatitis B, hepatitis C, and so on. See, some of them maybe we can avoid actually. If see, especially environmental pollution can be avoided. And here I would like to say, Honorable Prime Minister Narendra Modi brought the Swachh Bharat. And if everybody follow Swachh Bharat, and we can control less small portion of environmental pollution. Of course, there are so many factories and other things. They need to work on those as well. And for the lack of physical activity, I will just show one example. Of course, everyone is not in that, but you see here, see if most of the families around us, we know, watching TVs after the dinner, not doing any physical activity. This is one of the scenario one can stop. So, okay, now enough of the advices, but we will see what are the options, what are the treatment options available right now. One of them is radiotherapy, as most of you know. So radiation therapy is often abbreviated like this, RT, whatever, RT, RTX. Radiation therapy may be curative in number of types of cancers if they are localized to one area of the body. So this radiation therapy is only prevent tumor recurrence after okay, so radiation therapy will be used to prevent tumor recurrence after the surgery to remove a primary malignant tumor, for example, early stages of breast cancer. So radiation therapy is synergetic with chemotherapy. So most of the time radiation therapy used combination with the chemotherapy, and as is been before chemo or and during and after chemotherapy. So we'll go little bit detailed into chemotherapy here. So treatment options, chemotherapy. Chemotherapy, we can call as CTX as this abbreviation. Type of cancer treatment that uses one or more anti-cancer drugs, chemotherapeutic agents as part of standard chemotherapy regimen. Chemotherapy may be given with a curative intent, which almost always involves combination of drugs. So it will aim to prolong life or reduce symptoms. But so still we have the drugs which is prolonging the life or reducing the symptoms, but we need the complete cure. So I will go a little bit details into chemotherapeutic agents, which also called cytotoxic agents or anti-cancer agents. And these are the already known in the literature, all these several compounds like a doxorubicin, cisplatin, paclitaxel, so on and so forth. They are there in the literature, but they are all comes with some side effects. So none of them are without any side effects, which is common. So I will show some of these structures of selected ones like a mitoxantrone, paclitaxel, gemcitabine, vinblastine, docetaxel, ipothiolone. You may notice there is one inorganic compound in this anti-cancer drugs. There are several, but mostly organics. But this is the cisplatin is special. So my point being here, these are all used in right now for cancer treatments, various cancers, but they comes with the side effects. And there are some more compounds like a mitomycin C and tubulysin hydrazide. They are much more cytotoxic, very powerful cytotoxic agents. So problem with these agents is you cannot administer these to the tumors. So because since they are powerful, they can't differentiate between healthy cells as well as tumoric cells. It will also affect healthy cells. So what happens is it will cure the tumor, but in the meantime, your side effects are much severe. So you will have trouble surviving the patient. So what can we do for this one? Can we use these powerful agents in the cancer treatments? And if you know, if you see the defense situations, how military or defense people is designing missiles to precisely attack the target. For example, um, a laser guided missile. Laser guided missile uses a semi-active laser guidance to strike a designated target with great accuracy than an unguided missile. In US, in Vietnam War, they developed a laser guided missile and quickly proved their value in precision strikes of different point of targets. And this, of course, now worldwide, we have several technologies, modern technologies have a laser guided weapons. These weapons use on board electronics and track targets that are designated by laser, typically in the infrared spectrum, and adjust the glide path to accurately strike the target. Here is example. So you point the target and precisely guided, controlled, laser guided missile launched, and it will attack only here. So why I'm showing this? Can we do similar? Here is the enemy is here, but in the cancer, enemy is the tumor. Can we precisely target the tumor cell without affecting the healthy cells? So that's where this kind of thinking comes into the picture, which is well knownly called as a targeted drug delivery. So now I will go into the now targeted drug delivery portion of the cancer therapy. So I will call novel warheads. Warheads is basically take the anti-cancer agent attached to the attached to the um, linkage such a way that it will go directly precisely to the target tumor cells without affecting the healthy cells. So here is a design concept. So in the picture shown here is this top portion of the red is the drug, which is a highly cytotoxic drug, and the bottom portion is the ligand, and it will be connected to the hydrophilic spacer as well as the releasable linker system. This green portion is very important. So we have to stitch a molecule, anti-cancer molecule, such a way that this linker system will be released in the cell. After reaching the cell, it will be released. Until that point, it will be intact. As long as this part is intact, the drug will not harm any cells. So that is the idea. So here is the thing, and of course, my ligand here is the folic acid, and because these are the very specific to folate receptor positive cancer cells. So this is the ligand portion, and this is the drug portion, and connected by this spacer, which gives the hydrophilicity of the molecule like a water solubility, and this is the linker system green portion, which is a cleavable in the cell. It can be in endosome or lysosome. So this is the strategy. This will cleave in the endosome with this green portion and giving to this folate separated by drug. And this drug now is very powerful since it's cleaved in the endosome, and it will kill the target tumor cell. So here how it works. This is a cell. If you consider this is a cell, there is a nucleus, endosome, and lysosomes, and these are the folate receptors on the tumor cells. And the synthesized molecule you will send directly goes to the cell without affecting the other parts of the body, and it binds to the receptor and internalizes to the cell, and in endosome, and it will form the late endosome state. It will segregate all the ligand and drug separated, and drug will go to the nucleus and kill the target, and ligands will escape from the cell and recirculate. So this is the mechanism. I hope I kind of explained how this works. And here I showed earlier these anti-cancer drugs, for example, this mitomycin or tribulizin B and this desestelvin blastin hydrazide are very powerful, as it is this cytotoxicity of these molecules, especially in folate receptor positive cell lines. These are the IC50 values. So one thing we have to remember, after making the conjugate, this IC50 should remain same or similar, so that it activity remains, and the cleavable linkage should be constructed based on the endosomal properties. So these are the three of the conjugates with the mitomycin, vinblastine hydrazide, and tubulysin. And these are the parent drug IC50s. Conjugates IC50 is very within the ballpark of the drug parent drug. You can see 30 versus 36 nanomoles, and 2.4 versus 5, and 1 versus 1.5. That means these drugs are um, can be considered and taken into the clinical studies. And I will show here some more examples which we synthesize in the lab with using this technology, this design. And here is the another molecule with the mitomycin C. I will not go into details of synthesis. That will take a lot of time. But I am just giving overall picture what kind of molecules one can synthesize in the laboratory in organic chemist hands. And this is vinblastine hydrazide with this hydrophilic spacer in between with the ligand. And this is the epothelone, one of the another very potent cytotoxic compound. And this one is a tubulysin bihydrazide conjugates. These are the one drug connected to this system. And the organic chemist can go crazy. So can we put two cytotoxic compounds in one construct and send two cytotoxic drugs at the same time? That comes here, like yes, we can. And with the ligand, the same spacer we can have connect to the two cleavable drugs. It can be same or it can be different. And here is the example. And this is the mitomycin C as well as vinblastine was attached to the same ligand with the spacer connection. So this one was one of the initial best construction of the organic molecule we made. And here is a brief, I will not go details, but brief synthesis, because so this is the monoconjugate. And if you see in the final product, there are two disulfide bonds are very susceptible for the reductive cleavage. So to make second disulfide bond in presence of first disulfide bond is very tricky reaction, and a lot of skill, expertise needs to this construction. And we I will not go into the chemistry details, but this nitropyridyl leaving group is very strong. So this reaction, it needs to be deprotected this sulfur, and reaction should occur within instantaneously, so that this disulfide bond is protected, and which we achieved. You can see the reaction mixture profile. This is the product. Starting materials will be one of them, will be here and here. So this is just to show how organic chemists can make these complex molecules. Well, we made several of these. I just show few more examples. Okay, this is another conjugate with the two same drugs attached to the spacer and the ligand. And of course, as an organic chemist, we think high. So, well, we made one drug conjugate and two drugs conjugate. Can we make three? So that will be another step of more challenge. So we designed this molecule with this is the ligand portion, and this is the spacer. This is one drug, tubulysin B. This is the doxorubicin, and this is vinblastine hydrazide. And of course, here we made third drug connected by click chemistry. I'm sure, hope you know the click chemistry with the alkyne reacts with the azide, making this 5-membered ring. So we made this compound. Here, of course, any organic chemist, of course, here the intention is to make the medicines. That is, of course, that is most important. But as an organic chemist, how complex you can go? Can you make mono conjugate, bis conjugate, tris conjugate. This is a triple drug. So when we made this one, any organic chemist, what is the best day of the life? When you achieve something, when you synthesize challenging molecules. So I'm proud to say this triple drug conjugate was synthesized by me single-handedly, and that is the day. Actually, I can say it is my festival. It's like I can say Deepavali or the Dasara or whatever. So I am very happy when I made this compound, and this compound is in the preclinical evaluation, and hopefully one day this will go into the clinic. And of course, organic chemist will never stop their successes, or their um, they will they plan for more challenging things. And of course, in this case, since this is the first triple drug conjugate, we made some more. And of course, this in this particular case, we put the same drug 3 times instead of 3 different drugs. So this will be a little bit easier than the previous one. So because we took the same drugs instead of 3 different drugs, and we tried to put all 3 in using the click chemistry. And here, of course, 3 disulfide bonds were pre-made in pre-synthesized in one unit and attached these in one step. And this is the similar compound here. Sorry, here instead of disulfide, we used this Valsit Paba construction, and I will not go into details. Only thing I will tell you, this will be cleaved in the endosome using the enzyme called cathepsin B. So this will be, so whatever we plan, we have to plan such a way that it can be cleaved in the endosome. So this is the cathepsin B cleavable Valsit linkers, and connection site is very similar. And here is the final thing. This is the triple drug, but it is it's much more challenging than the previous one because of this um, different kind of connection. Because sorry, see here, uh, we use the same Valsit Paba cleavable linker using cathepsin B, but we just put the amide bond in various reasons, but this is much more complex than previous ones. And when we made this, this will be like a special achievement for organic chemistry. So here, I narrated not only targeted drug delivery, so how organic chemist can synthesize such a complex molecules. These molecular weights and this number of carbon atoms is more than 100 carbons. Um, so as an organic chemist, I am very happy to be part of this molecule synthesis. And I will say, whatever the new molecules I presented today, 80% of them are synthesized in my hands. And hopefully there are few of these compounds, what I showed, or at least one of them will be future medicine, going through all the steps involved in the drug development process, as Ravi narrated in his previous lecture. So with that, I will give some uh, brief summary. So basically, I went through the simple introduction to organic chemistry first to understand, to just showcase to the uh, uh, early students to understand, from the bottom level. And then I showed brief synthesis of Golcondian, which is my part of my doctoral study. And then I went through the organic chemistry in everyday life, what we eat, what we live on, and how the regular medicines are involved in by organic chemistry. Then went to the introduction of cancer and causes and treatment. And then finally, I showed targeted drug delivery designs of SMDCs, mono and dual drug conjugates of cancer therapy. and systematic approach of triple drug folate conjugates were highlighted. That is one of my favorite synthesis have done so far. And finally, I thank you for Palamuru University giving me this opportunity to present my brief talk, and especially Professor Girija, my colleague at University of Hyderabad in Masters, and of course arranging the. I want to thank everyone. I may not go everyone, but Vice Chancellor Rahul Boja, Mr. Garu, and Professor Pawan Kumar Registrar Garu, and Madhusudan Reddy Garu, and Chandra Kiran Garu, and Sudarshan Reddy and Gnaneshwar. Thank you very much. And all others, organizing team, technical support, I will not go through them. And finally, thank you for your attention.
Thank you, sir. Thank you for your scintillating lecture on significance of organic chemistry in medicine. Now I request participants, as a discussion is open for you. If you have any questions, you can ask Hari sir.
Hello, sir. This is Ravi. It's very interesting presentation, and it was. I can understand how challenging those triple conjugates and synthesis of those molecules, and also I can relate to one of the experiences you said about the Diwali. Like when you hold the first NMR, the happiness in our faces.
Oh yeah,
So,
yeah, sure.
Yeah. And these triple conjugates, how are they formulated?
Well,
because it's those big molecules, the solubility issues, and
yes.
how are they actually formulated?
Well, of course, I know this will be very challenging to CMC, but of course, I am not involved in that. But especially the triple drug one is still in the process. But dual drug, one of the dual drug is very similar to the similar complexity as triple drug. And one good thing is these are water soluble compounds, because that's how we design the spacers. That part is good, but only thing is stability we have to make sure. And of course, I am not involved in that process, but I know. So sometimes, final purification is prep HPLC purification. So we will use the buffer to stabilize the molecule, and then freeze dry. So once we freeze dry, in most of the times, before freeze drying, we have to desalt the buffer using the prep purification. But I don't know the finer details of the formulation process, but these compounds can be lyophilized and stored in the clinical before, especially clinical samples. Once you purify before lyophilization, you can put it in the clinical vials and freeze dry in the clinical vials directly. But but it will be challenging for the CMC department. I can guarantee that one.
Yeah, that is for sure. Looking at those molecules,
Yes.
Yeah.
Yeah. And I am not involved in that, but I have seen, because in my CMC department, I closely associate with them in the technology transfers. Sometimes they have issues, I have to help them. So, but it will be challenging, but water solubility helps. We don't need any special formulations like sometimes molecules which are not soluble in water, you need to formulate a little bit differently.
That is correct.
Yeah.
Especially not water solubles, you may have to go through the lipophilic formulations or some other techniques to deliver the acid.
Yes.
Is it
Any
over,
other questions?
Hari?
Yeah, I am done. Any other questions? I am willing to spend some time.
Are you related to Damodar sir?
Damodar Reddy?
Yes.
I am my own brother.
Oh.
I am younger brother. I am younger brother to Damodar Reddy. Yes.
Okay, okay, yeah. That's why, like, I could relate the facial features and, like, I was like wondering, like,
Yeah.
okay,
Girija knows about it, but
Yeah.
yeah, I am younger brother. We are four brothers. I am the youngest, and
yeah.
I am
Actually,
organic
like,
chemist.
right before I came to US, like, I worked in MNR college, like, with,
Oh, I see. Okay.
yeah, I worked as a lecturer there for, like, taught organic chemistry there.
Oh, I see.
And,
Okay.
and
Okay.
Ravi, in fact, Damodar Reddy worked even in Navy
Yeah,
College, but I
yeah, he
think you
was,
left
yeah.
then. By then, I
No,
think you have left. Yeah.
no, madam. I think he was there during my first year. He taught inorganic chemistry there.
Inorganic chemistry. Yeah.
Yes.
Yes. Yeah.
Mr. Hari, I would like to.
Hello.
Hi,
Hi.
Sharma.
Hello.
Good
Yeah.
evening.
Hi,
Good
Sharma.
evening. I'm really wondered. I could see the other part of Dr. Hari Krishna Reddy. Now I don't want to call you Mr. Hari because though I have the intimacy with you, I could see the intense work you are extending in the field of organic chemistry and medicinal drugs. The way you explained the challenge what you presented in the beginning and the way you explained the difficulty in achieving it and that hydrophilicity and all that linkage and all I could understand being a chemist but you have grown to such a big extent involving in the global challenge and I know your committed work towards chemistry right from your 20 I mean I should say 21 years age now that you are you may be somewhere around 55 56
Yes,
but
I am
the
55.
way you presented it could reveal the hard work you are putting in making it realized in the drug field and definitely all your endeavors will come true the mankind human what kingdom will be definitely blessed by the effort what you are putting the universe will definitely set its equations towards your work. Thank you, Hari. I'm so proud of you. I'm
Thank
very
you.
happy.
Thank you, Sharma. I
I'm
am also hoping.
overwhelmed. I'm seeing a new Hari now. Very
Well,
happy. Thanks you, Girija. Thanks a
Thank you.
lot.
Thank you, Sharma. In fact, I should thank you because you have logged in and you have rightly said about Hari. Actually, I wanted to say all this, but you made my job easier by
I'm
really.
so
Yeah,
happy. So
yeah,
happy.
really.
yeah.
Thank you.
Thank
I
you,
don't
Sharma,
want to
thank you, Sharma, attending the seminar because it's.
come on.
I
Come
know.
on.
Yeah.
It's very valuable.
Very
Very
valuable.
valuable.
Yeah,
So I don't want to
yeah,
waste the time also. Thank
yeah.
you.
Okay then. Thank you, Sharma.
Thank you.
Yeah. See, the concluding remarks, coming to the concluding remarks of today's lecture given by Dr. Hari Krishna Reddy. So I'm very happy to say in the beginning because reason is he was my classmate and at PG when we were doing MSc. We used to share how we used to share even in MSc at MSc level. We used to wonder how Hari and his group like Jaipal Reddy and another Balkrishnan Raju when we used to do lab in the lab, we always used to get very low yields. We used to have marks allotted even for both yield as well as the purity of the sample, and we always used to wonder how these people could get so much of yield without affecting the purity of the sample. Normally, if we get the yield, we miss the yield, and miss the purity, and if we have the purity, so it's both are not connected all the time. So we used to wonder, and we always used to ask these people, Hari and Jaipal and Balakrishnan Raju. Then they used to uh, they used to pull our legs and they used to give us. Suppose if you don't have the compound in the final submission, they used to share some of them. They used to give us, and that's how we used to. And sometimes even when we were in MSc, when we never used to understand the basic organic reactions, they used to explain. And we all used to sit in the library till 12 o'clock when we were in MSc before during the exams. And I can understand how he used to explain us. And later on, he completed his MPhil in Dr. Baswaya's lab. And after that, he pursued his PhD under Professor Govardhan Mehta. And it is not that easy to complete PhD under his guidance because 6 years means it is almost 12 years you can consider because right from 8 o'clock in the morning till 12 o'clock in the night they used to do, and he took 6 years, 6 long years. And we always used to wonder, especially the Mehta's lab, they used to have all these what the molecules, fullerene molecules, and how they are fascinated, how they are fascinating the chemists. So he explained about the synthesis of Golcondian, which in fact such complex molecules, even though they don't have any specific biological activity, but still they're initiating the chemists because of their complexity, and they're fascinating the chemists to synthesize. So in fact, when he explained, even though that molecule, the carbon is sp3 hybridized, the bond angle or the dihedral angle, as he said, is not 109. It will deviate from that because of the strain, but still once it is synthesized, by looking at the molecules, we think that they are very difficult to synthesize, but once they are synthesized, they have high stability, and this is what we discuss under topological studies. So definitely I just could get two of the reactions when he explained about Golcondian, because we didn't go to the details. So 2+2 cycloaddition reactions and Wolff-Kishner reduction. They look very simple, and we think that they're very simple reactions, but it takes so much of years, so much of time to synthesize that molecule. So it took almost 6 years to synthesize such a molecule, and later on he started, how the way he has started his lecture, that shows his simplicity. Even though he is such a big person now, and as Sharma said, yes, he is different when he was in MSc, and now he's totally different. It is really, I can't imagine his position over there. But still, it shows the way he presented the lecture itself shows that he's down to earth, simple person. He started with carbon valency. Generally, the people who are going to give a talk, especially with regards to their research in such international webinars, they directly start with the topic. But he didn't do that. He started right from carbon valency, and then he slowly came to the class of compounds, alkanes, alkenes, based on the bond angle and the hybridization, and slowly he talked about the reactions, different types of reactions, and the stereochemistry involved, and then slowly he's introducing he introduced ourselves into the topic, and then he talked about the organic chemistry reactions, how they are important, and what are the organic molecules which are used as medicines, and then finally uh, how, what is he actually doing, how the organic molecules, some of the molecules are used as drugs, especially in cancer treatments, and what are the already available drugs which are used in cancer treatment, and what are the side effects, and then how actually these people are doing, what exactly they are doing by targeting the only the cancer cells, without affecting the healthy tissue, using the principle of military people, that is targeting laser guided missiles. So based on that, they thought a lot about it, and then slowly using linkers, and then ligands, and then the breakable linkers, and through spaces, how they have linked the ligand with the drug molecule, and how it is released into the site. So only it will be released into the only the diseased cell, the site, so that the healthy tissue is not affected, and how he developed the dual drugs and the triple drug conjugates, and really it's wonder. And as he said, definitely the chemists are very crazy, and they're not especially, as he said, organic chemists, they don't stop at that one particular stage. So they keep on moving, and people like Hari, definitely God should give them longevity, good health, and definitely these drugs, as especially as he said, this particular drug what he has developed, they're going to be marketed later in the future to treat cancer, because cancer is the deadliest disease. Even though we say there are so many treatments which involve radiotherapy and chemotherapy, still people are suffering, and many people are dying due to different cancers. And hope this particular drug is going to be marketed in the near future, and the whole mankind will be benefited. And then once again,
Madam, it's not audible.
it's not
No.
okay. One second. Okay.
Yeah, now it is good.
Yeah, is it okay? I got a phone call. That's the reason why.
Okay.
So where did I stop? I want to complete say that. Where did I stop exactly, Hari?
Well, you are talking about the marketing, the future marketing of my compound,
So these
etc.
drugs, because they have put in so much of effort, and this drug later on, in the near future, Definitely, we hope that it will be a better medicine because of the side effects, less side effects. And as I said, any medicine definitely for that matter, they'll have side effects. And but because this particular, based on the mechanism what they have developed, the technology, it will be targeted only at the site. So naturally, the healthy tissue is not disturbed, and definitely it becomes a frontline. These medicines would become frontline medicines for the cancer treatment. And I think all the participants are benefited by his talk. And I once again thank you, Hari. Uh, thank you very much. And once again, he's also from Mehboob district, and so that is one more pride moment for all the Palamuru University students. So I'm very happy that all the three out of four speakers, all the three speakers, they hail from Mehboob district. And thank you,
Actually,
Hari.
your village should rename it like Golconda and like something.
Yeah.
Chemist in
You're
inside.
there.
Your village has produced as well so many great chemists.
Yeah,
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 yeah.
really.
Actually, yes, as I was telling earlier with Ramu Yadav and Dr. M.S. Sen is my father's brother's son, like my Baba, Mena Baba. Mena Baba means, so
Yeah.
we are like a neighbors in my group, my village. Actually, of course, MSN is, you all know MSN is
Yeah,
how,
MSN Reddy తెలుసు మాకు.
yeah, yeah,
మాకు
yeah.
చాలా కాంట్రిబ్యూట్ చేశారు. Actually, when we have conducted for the first time in 2015 national level national seminar, he contributed 1 lakh at 2015 itself. He contributed 1 lakh as a donation to the seminar.
No,
Yeah, sure.
forget about Mahbub Nagar produced so many. Hari's family itself produced
Yeah,
how many PhDs, Hari?
Three PhDs.
3 PhDs
Damodar,
out of 4.
yourself.
My elder brother Narsimha Reddy.
All the three are doctorates. I've just fallen on their mother's feet. Once I could
Yeah.
I was fortunate enough to see that his parents in Chilkuru temple. I just fallen on Hari's mother's feet that day because the three doctorates mother we rarely see. That too, they are from chemistry. So which family can be a better representative of chemistry than this?
Yeah, yeah.
That too to a very high extent. It's
Yeah,
not
very
casual,
high.
this thing. So happy. Thank you so
Thank
much.
you, Sharma. Thank you. Thank you
Thank
for
you,
your.
Sharma, for your presence, and not only that, giving remarks and compliments, and then talking about him on this platform.
Thank you. Thank you all. Thank you. See you. Thank you.
So good day, Ravi and Hari, both.
Yeah, thank you.
Madam.
Yeah.
Good day. Good day for you.
Yes.
Thank you. We need your support further, Hari.
Yeah, sure. Definitely. In any ways, whatever. Whenever of course, I don't know when I will visit Mahbub Nagar now because of these pandemic
Yeah,
situations.
pandemic.
But when I come there, and we will meet you again, and then even online anytime, we can have a discussion with your students or something. I think we can have informal discussion with the students in Zoom meeting.
Yeah, we'll arrange, Hari. We'll arrange that.
That will be.
And now, Sudarshan, before I forget,
Yes, madam.
the appreciation certificate. In fact, I forgot.
Yes,
I totally forgot. A certificate of appreciation to Dr. Hari Krishna Reddy for his outstanding presentation.
Thank you.
Congratulations.
Madam,
Thank
one
you.
of our participant would like to express his feelings, feedback on this teaching.
You want to? Okay.
Yeah, no, Shivakumar Singh, sir.
Shivakumar Singh. Okay.
Yes. Sir, you can unmute.
Sir.
Good evening, all. Madam, really, it is the one of
good evening.
One of the wonderful program, madam. Within a short time, the Department of Chemistry, like you eminent people, have been organized like these types of program. After listening all the talks, I should say definitely Palamuru is not rural background area. See, sir, on those days only, like you eminent personalities have been from this particular area. And Madam Professor Girja Madam has told, like eminent personalities having started from fundamental onwards, sir. Because I am particularly from biological background. My PhD on natural products, and my missus also doing on organic chemistry, sir. So really, like Ravi sir and Hari Krishna sir and Ramu sir, and, and I also observed another one thing here, that uh, besides of the subject, we should have, we learn many more ethics, ethics in the studies, sir. Like teacher and student relationship, that never ends. Like many things we have learned, because these things required in upcoming researchers and upcoming teachers also, how to maintain the relationship of the student and teacher. Like really, these things really required today. We learn subject and as well as these ethics things. So on behalf of our department and as a participants, I grateful to the eminent personalities, Krishna Hari sir and Ravi sir, Ramu sir, and the morning session just have lately joined. But the evening session also very much informative. And I thankful to Professor Girja Madam and Dr. Noorjahan Madam Garu and Dr. Chandrakiran sir and other organizing members, each and every faculty member of the chemistry department. Really, sir, Professor Girja Madam always encouraging the upcoming researchers. As I have seen, like Ravi sir, after discussing of the student and teacher relations, really we have been very much learned and attracted, sir. On behalf of myself, I really are grateful to the professors and eminent personalities. Thank you very much as a part of this program. Thank you, madam. Thank you, madam.
Thank you, Shivakumar.
Madam, Madam, there is a question in the chat box for Hari sir. Just I will read out.
Yeah.
Sir, Hari sir.
Yeah.
If we use Golconda with protein and lipids or fats as solvent,
okay,
does it work as a cancer drug? Is there any possibility?
No, it will not, because it is there is no functional groups on that, so it will not. Only that particular molecule, as I mentioned in one of my slide, if you put the polynitrations, it can work as a propellants, because there is no functional groups at all. As it is, it will not work. Of course, you can plan for function derivatizing. That will be more challenging once again. So yeah, answer is no.
Okay, sir. Thank you, sir. Now I request to Professor K. Girja Madam to propose vote of thanks for today's international webinar. Madam, please unmute.
Unmute.
Yeah. Yeah.
Yes.
Sorry. So we are closing the session today, and even though it is an international webinar, because of this sudden commencement of the exams for the PG students, we have confined ourselves to one day international webinar. And first of all, let me thank our in charge Vice Chancellor, Dr. Sri Rahul Bojagaru, and the Registrar, Professor Pindi Pavan Kumar, and Voice D, Madhusudan Reddy, for constant encouragement and support to organize such webinars. And then coming to the esteemed speakers and all the participants, in spite of their busy schedule, especially the speakers from IIT Delhi and both Ramu Yadav and Dr. Ramu Yadav and Dr. Janki Ram Vaitla, and two speakers in the evening session, Dr. Ravi Kumar and Dr. Hari Krishna Reddy, for their valuable lectures. And not only that, paying so much of attention to and listening to all the lectures. Hari actually started right from 4 o'clock, and Ravi also waited till the end, and they have shared all their experiences as not only as a research scholar, but also after joining into their respective institutes, what are the researches going on, and then all other technical support and the colleagues in the department and the integrated chemistry department, the technical support, especially Arjun, Dr. Arjun, and the other his supporting team. And just now, Shiv Kumar Singh, he logged in. He shared his views, and all others. If I forgot anybody, and even the other support, Mallikarjun, technical support, Mallikarjun, and all other participants, especially for patient listening right from morning. In fact, the session started today in the morning around 11 o'clock, and then it extended up to 7:30 now. But still, all are patiently they're stuck to this, and then they have listened, and they made, in fact, they noted all the important points. And thank you all for making this a big success. Thank you once again, all my colleagues in the department, without whose cooperation this wouldn't have been a great success. Thank you all. And my colleague, especially Dr. Noorjahan, and she, in fact, she is behind this. She made it into, actually, she's the one who materialized this. In fact, we thought of planning this. We planned before, like 2 months ago only we have planned because of the COVID. Actually, initially I was affected with COVID, and later on Noorjahan was affected with COVID, and the whole family was affected with the COVID, and that's the reason why it got postponed, and suddenly the exams commenced. But in spite of that, she took a lot of pains in making it a big success. And again, the two juniors in the department, even they helped us a lot. Thank you once again, all of you, for patient listening. So thank you.
Thank you. Thank you.
Thank you, madam,
Thank
for
you.
your vote of thanks and nice words. It is you, being the senior, who continuously encouraging us to do such programs. Being senior, you never showed that we should obey you or so, but you always been along with us and working equally with us, and continuously you are there right from the beginning and till the end, and you are materializing the things and communicating to the speakers and everything. Not only me, you are equally have the credits in the success of this webinar. Thank you very much, madam, for
Thank you.
extending your
Thank you.
help and cooperation.
Thank
And
you.
thanks all the participants and the speakers for being with us in this virtual international webinar. And we look forward for many more coming programs virtually. So thank you once again for your kind support and cooperation. Thank you.
Thank you. And thanks once again to Shiv Kumar Singh. Really, you spoke well, and I'm very happy that you got connected.
Thank you, madam. Thank you. It's my pleasure,
Thank you.
madam. Thank you.
Yeah, thank you. We conclude now. Thank you.
Yeah. Thank you, Girija.
Thank
See you.
you, Hari. Yeah.
Thank you,
Bye.
everyone.
Thank you, madam.
Thank you.
It
Thank
was
you.
pleasure meeting you, Hari.
Yeah.
Sir.
Ravi, yeah, thank you.
Yeah.
And we'll try to keep in touch, I think.
Sure, definitely, sir.
Yeah,
Thank
sure.
Thank
Okay, see you all.
Yeah.
you.
Nice. Thank you. Bye, Noor.
Bye, madam.
Bye. ఇంటికి వెళ్ళి ఇంకా పాపం పొద్దునుంచి ఇక్కడే ఉన్నట్టున్నావు.
Yes, ma'am.
Thank you, Ravi, and Gnaneshwar and Sudarshan also. All others. బాలరాజు కూడా కనిపించాడు ఇందాక. All the take. Okay. Bye. Thank you. Bye. Bye. See you.
Thank you, everyone.
Yeah.